supply primarily nitrogen for protein quality. So I have a field experiment that's looking at planting data nitrogen supply, and then I also have a growth chamber experiment looking at nitrogen supply specifically, and then looking at the nutrient quality in the seed. That's so awesome. Mostly just trying to tease out how stable are those qualities across agronomic differences, even within the same environment.
Awesome.
So, all right, I'm just gonna go through. So yeah, we have about 5 minutes before we're gonna get started. So again, if you need some water, go to the bathroom. Sounds like we don't have to do a mic check. Everyone seems like their technology is working, so it should be fun. And then, hello, Sonia.
Hello. I'm ready.
Hey, Sonia, I have my Spanish glossary of plant pathology terms pulled up in case there's any weird translations you have to do.
Okay, thank you.
Yeah.
And can you share it with me?
Yeah, let me get that sent over to you.
Thank you.
I'll just put it in the Q&A, I think.
Sure, or you can, I think you can send like a private message too.
It's blocked for me.
And
also it won't let me do a question.
Maybe if you email me, I can forward it to Sonia right now.
That works.
There.
Okay.
Sorry about that. And then Evan will get started at 10:05. Does that sound good to you?
Yes.
Yes, see.
All right, Rachel, you should have it.
Good morning, everyone, and welcome to the third day of the International Quinoa Research Symposium.
Muy buenos días a todos y bienvenidos a nuestro tercer día del Simposio Internacional sobre la Quinoa.
This is the first session in Room 2, Horizons in Quinoa Production Systems, Breeding and Pathology.
Esta es nuestra primera sesión en la sala 2, llamada Perspectivas en Sistemas de Producción de Quinoa, Mejoramiento y Patología.
If you're looking for Room 1, the heat stress presentation, you can find the link to that Zoom meeting in the Slack channel.
Si ustedes están buscando la sala número 1, pueden dirigirse al canal de Slack y ahí van a encontrar el enlace para la presentación que tiene que ver con estrés por calor.
Here are some tech tips for participants in this session.
Aquí tenemos unos consejos técnicos para los participantes de esta sesión.
Please use the Q&A box for questions during the presentation.
Por favor, utilicen la caja de preguntas y respuestas para que escriban sus preguntas durante las sesiones
You
o
can
las
also ask,
presentaciones. Perdón.
gracias. You can also use the Slack channel to post questions, and any unanswered questions will be posted to Slack for the presenters to answer after the presentation.
Muy bien, también pueden usar el canal de Slack y recuerden que cualquier pregunta que no pueda ser respondida en vivo va a ser respondida en nuestro canal de Slack y ahí más tarde podrán encontrar las respuestas.
So now I will hand things over to the session facilitator, Rachel Breslauer.
Pues ahora le voy a dar el mando a nuestra facilitadora, Rachel Breslauer.
Thank you, Evan.
Gracias, Evan.
Welcome everyone to the Horizons in Quinoa Production System session.
Bienvenidos todos a Perspectivas en Sistemas de Producción de Quinoa, Mejoramiento y Patología.
My name is Rachel Breslauer, and I'm a PhD student in the Sustainable Seed Systems Lab at Washington State University.
Mi nombre es Rachel Breslauer y soy una estudiante de doctorado en el Laboratorio de Mejoramiento de Semillas de la Universidad Estatal de Washington.
Increasing global acreage of quinoa is contingent upon advancing quinoa breeding and plant pathology in novel regions.
El incremento, sorry Rachel, can you repeat it?
Of course. Increasing global acreage of quinoa.
El incremento del cultivo global de quinoa
relies upon advancing quinoa breeding and plant pathology in new regions.
Avanza a medida que avanzamos en el descubrimiento de patologías de las plantas en cada región.
Speakers in this session will outline their visions for how their disciplines can pave the way to expanding quinoa production globally.
Los presentadores que tenemos en esta sección van a expandir en estas ideas de cómo podemos abrir camino para las personas que trabajan en producción de quinoa, en mejoramiento y patología.
Dr. Andres Torres Salvador is the Managing Director for Radical Crops.
El doctor Andrés Torres Salvador es el director para el laboratorio.
The company is called Radical Crops.
Para la compañía Radical Crops.
It's a company that develops and commercializes saponin-free quinoa varieties suited for diverse climates, latitudes, and end uses.
Es una compañía que trabaja con saponeo de las semillas de quinoa.
We also have Dr. Anna Teston.
También tenemos a la doctora Anna Teston.
Dr. Anna Teston is a USDA plant pathologist.
La doctora Ana Testen es una doctora en plantas y patología.
Based in Ohio, USA,
En Ohio, Estados Unidos,
but has experience in disease detection and management in specialty crops in
pero
Ecuador. Oh, sorry.
okay. Doctora Ana Testen es fitopatóloga de USDA con sede en Ohio, Estados Unidos, pero también tiene experiencia en la detección y gestión de enfermedades en cultivos especializados
In Ecuador, Bolivia, Tanzania, and Ethiopia.
Ecuador, Bolivia, Tanzania y Etiopía.
We are very lucky to learn from Andres and Anna today.
Somos muy afortunados de aprender de Ana y Andrés hoy.
Thank you both for being here.
Muchísimas gracias a los dos por estar aquí.
We will now begin with a talk from Dr. Andres Torres Salvador.
Nosotros vamos a empezar ahora con una charla o con la presentación del doctor Andrés Torres Salvador.
Welcome everyone to the Horizons in Quinoa Production System session. My name is Rachel Breslauer and I'm a graduate student in the Sustainable Seed Systems Lab at Washington State University. And it's my pleasure to be your facilitator today. I would like to introduce our speaker, Andres Torres Salvador. Andres is the managing director of Radical Crops in the Netherlands. Radical Crops has partnered with Wageningen University and research to develop and commercialize saponin-free quinoa varieties suited for diverse climates, latitudes, and end uses. Since 2018, Andres has been responsible for initiating and supporting commercial partnerships with professional agricultural firms that have successfully established quinoa production chains outside the Andean region. Andres also focuses on the development of the R&D roadmap of the joint breeding program with Wageningen University and Research. He aims to ensure that development of breeding technologies and varieties align with the needs of quinoa producers worldwide. Andres is an Ecuadorian national and holds a master's degree in plant biotechnology and a PhD degree in plant breeding from Wageningen University and Research. He is an advocate of increasing biodiversity in agriculture in support of a more resilient global food system. I'd like to turn the rest of the time over to Andres, and I look forward to his talk.
Rachel, thank you very much for that kind introduction. Also on behalf of Robert Van Loo, who is the head of our breeding program and a co-author of this presentation. I also want to thank the organizers and the participants of the symposium for this wonderful opportunity. At Radical, we believe that quinoa can transform global agriculture. And today I want to tell you more about how we are building a modern seed company that can help transform quinoa into a global commodity. I always begin with some technical problems. I think I got it. Now, before I begin, let me give you some historical context because most of you are probably wondering, where did these guys come from? Radical Crops actually dates back to 2014, but you have probably known us as the Quinoa Company. The Quinoa Company started the first quinoa production chain in the Netherlands using the saponin-free quinoa varieties that were bred by Wageningen University and Research. In 2018, the Quinoa Company acquired the licensing rights to these varieties. And back then, our objective was to support the global expansion of quinoa by promoting the use of these varieties around the world and by building a global ingredients brand around them. But we quickly learned that the world actually needed a specialist quinoa seed company. So since the third quarter of 2020, we have stopped our quinoa production and retail activities in the Netherlands to focus on the creation of the world's first modern quinoa seed company, if I can call it like that. And our ambition is to continually deliver innovations in genetics and seed technology so that we can ensure that people have the best varieties possible, that we can increase the productivity of quinoa, and that we can diversify the industrial applications of quinoa. Understandably, with a new focus, of course, comes a new identity. And so the name Radical Crops perfectly embodies this ethos of our new company. We really believe that to build a resilient agricultural food and food system, we need to add more biodiversity to farming systems and global diets. And this requires making significant investments to adapt underutilized crop species to modern production systems. And yet you rarely see legacy seed companies do that. The radicle is the primary root of a seedling, and it signals the start of new life. And as a company, we basically want to tackle the biodiversity challenge at the root, and we want to give life to new crops by investing in breeding technology and innovation. So that is radical crops. And of course, you might be wondering, okay, well, how do you build a modern quinoa seed company? Well, Radical has partnered with Wageningen University in Research. Already I have mentioned that. Wageningen University in Research is the world's leading agricultural university. And it is also a pioneer in quinoa breeding, genetics, and agronomy with almost 30 years of experience in the crop. Together, we have created a very dynamic private-public partnership with specialist functions. Wageningen University and Research, they run a very agile breeding program which focuses on breeding sapwood-free varieties, and Radical Crops has the commercial exploitation rights to those varieties. And basically what we do is we focus on promoting their utilization around the world. Now, this organizational synergy has two very important advantages. The first one is that by building a global network of users of the varieties, Radical has created a steady income of resources, so a steady income of resources that can be continually reinvested into the program so that we can enhance our breeding operations. And also, of course, because we're in constant contact with the clients and the users of the varieties, we can provide the breeders with critical guidance on what the breeding program should focus on. So this is, of course, very powerful. That's the commercial side. Today, our breeding program employs 6 breeders and scientists, which are led by Robert Van Loo and Jason Abbott. Both of them are actually historical figures in the European quinoa breeding world. But what I most mentioned, of course, is that aside, in addition to that team of 6 people, they are supported by the university's specialist farming operations team. And we also have a wealth of amazing graduate students that continue to help us innovate in quinoa. So as you can imagine, the second great advantage of this public-private partnership is that we can run like a research and development powerhouse. Our close ties with the university give us access to a leading academic program in quinoa genetics and physiology, state-of-art technology and infrastructure that only a university can buy. But above all, Wageningen has been called the Silicon Valley of agriculture. We're basically at the epicenter of innovation in the plant sciences. So we are surrounded by an energetic talent pool that is constantly producing cutting-edge solutions to modern problems. It's really, really fantastic to work with these people. And of course, the breeders, Robert and Jason, had asked me to put a picture of them because they also want to be seen by the world. So here's a picture of who I'm talking about. And of course, they need to be recognized for all the amazing work that they have done. And so actually, this is a great opportunity for me to tell you a bit about what these 2 gentlemen have actually achieved in the field of quinoa breeding. Basically, for over 30 years, the primary goal of the program has been to deliver varieties that can perform at the highest level in modern mechanized production systems. The first step was to adapt quinoa to the production cycles of northern latitudes, and this basically entailed breaking the species photoperiod sensitivity, but also adapting the phenological stages of the plant so that we could capture effectively the European growing window. Now, the other thing that we did, of course, was to massively overhaul the morphology of the crop. The program focused on creating unicorn idiotypes with compact protruding heads and a very effective leaf habit. The goal was to perfect quinoa for mechanized production systems, but more importantly, the idea was to improve harvest index so that we could support a higher productivity in the varieties. And of course, the grand highlight of the breeding program was actually creating sapwood-free varieties. And so basically, even today, the cost of desaponification of quinoa can be an entry barrier for local production chains. So if you can preclude the need for abrasive polishing or specialized or investments in specialized equipment to desaponify, then you can make a much better case for starting a quinoa production chain. So this was really one of the major innovations from the breeding program. And so since the introduction of Atlas in 1999, which was the first non-sapwonin, sapwonin-free variety that was developed by the breeding program, Jason and Robert have delivered 8 additional sapwonin-free varieties. So we have quite an interesting portfolio right now. Let me just quickly check something here. Basically, I don't know why this is not working. My apologies for this. There we go. So basically, the portfolio has 2 very important characteristics. The first one is that we have created a lot of variation in crop cycle length. So you can really see that we have early, mid, and late varieties. And basically what this does is this creates opportunities for growers to capture different growing windows across the world. So you can really go to different parts of the world where you have maybe shorter growing seasons or maybe longer growing seasons. The other important characteristic of this of this variety portfolio, of course, is that we have continually been improving on yield. So if you can compare here, I don't know if you can see Atlas, that was our first variety. If you compare it with Duchess, which is one of our latest varieties, you will see that Duchess can actually have 25% more grain productivity. Of course, this is when both genotypes can complete the growing cycle without major interruptions. And so one of the things that I do want to point out here is that what we have tried to achieve with this set of varieties is that we have really tried to improve the grain productivity of quinoa. Today in Europe, the varieties, the realized commercial yields, so I'm talking about, you know, actual producers' fields, has surpassed 2 tons per hectare. So this is actually pretty good. And actually, when you look a bit deeper into the numbers, you will see that there's quite some variation between farmers' fields. So some farmers can actually get anywhere between 1.5 to 4 metric tons per hectare. And so this is very interesting because, of course, climatic conditions determine what you can do with the varieties, but it's also a bit of the farmer experience. So if you have more experienced farmers, you can almost certainly get a stable 3 to 4 tons per hectare with these varieties. And this is something that we actually see in Chile. The South Pacific Seeds in Chile, it's a great company. I don't know how they're doing it, but they are really pushing the varieties to their frontiers. And it's pretty amazing to see because they're radically changing the world of quinoa production. Again, there we are. And of course, one of the other things that we have tried to achieve in this in our variety portfolio is to create more characteristics also or more different characteristics in grain quality, grain sensorial quality, grain physical aspects. Here you can see that we have tried to make a black variety called Calyx D. We also have a variety called Rouge Marie, which has red grains. The always elusive, bigger, wider grains in the market. They're always hassling us about the color of the grain. So we've actually tried to create something that looks a bit more like South American quinoa. And of course, we're also working on more the industrial quality aspects of the grain. So if you look at Riobamba, my apologies. If you look at Riobamba, basically has neutral aroma in bakery products. So this is quite an interesting variety for millers. So you can also see here the range of varieties that we have from Calyx D to Jessie. And so today, basically the varieties, these varieties have enabled the establishment of successful quinoa production chains in over 20 countries across 5 continents. This year alone, Radical supports 5,000 hectares of quinoa production, and we expect that our partners will aggregate about 10,000 metric tons of commercial product, which is pretty nice. Most of the production today concentrates in Europe, but we are beginning to expand fast in the Americas and have some really promising projects as well in Asia, Oceania, and Africa. So I'm really interested about those as well. And basically, the way that we support our partners is that we extend to them licensing rights so that they can use the varieties, they can create controlled production chains. We also support them by providing them superior sowing seeds. So we really want them to get the best start that they can possibly get with the varieties. And of course, by having a network with so many partners, we can collect experiences and provide agronomy support within the network. And this is very important because what you want in the network is that everyone can do or can push the varieties to their highest potential. Yeah, so this is the current state of Radicle. This is what we're doing together with our partners in Wageningen University and research with the current varieties. And it begs the question, what is there in the future? So what do we try or what do we want to achieve in the future? And if you ask me that question, I always say, well, it's all about increasing the productivity of quinoa. Like I said, we basically want to turn quinoa into a global staple. And to be competitive with other commodities, basically the realized productivity per hectare of quinoa needs to radically improve. Now, crop models that we have developed in-house show that our breeding program can deliver varieties with a yield potential of 5 to 6 tons per hectare. In fact, ongoing selection work in the breeding program already shows that we can surpass the productivity of our current varieties. For example, if you see in this graph, relative to the productivity of Atlas and Duchess, you can already see that some of the selections that we're making this year and that are on the fields right now in pre-commercial testing can do much, much better in terms of yield. So we can get there. But of course, what's very important to realize is that we still need to close this yield gap because in the producers' fields, the results are just not at that level that you want them to be. So our work today basically focuses on developing varieties which have the ability to mitigate a broad range of stresses across the growing season so that we can guarantee the highest yield potential of these varieties. In other words, we want to ensure that these varieties are dependable and predictable in farmers' fields, not just in experimental plots. So this really means that we need to somehow fast track quinoa to the 21st century. We need to treat it like you would treat maize or wheat, and that requires a lot of investments in technology. I hope we can edit those things later on. But yeah, so what I can tell you right now is that I'm very confident that Radicle can get to this vision of a quinoa that has been commoditized. Basically, I think that we have all the right ingredients for it. As I have hopefully shown you in this presentation, I think that the breeding program already has some exceptional elite lines that can be the basis for our future breeding program. And you know, there's also a network effect with our partners because we're basically extending our testing capability so we can really look for opportunities to look to improve on yield stability. So the next logical step for us is to scale up and to make our breeding program go faster. And last year, Radicle and Wageningen University were the recipients of a grant from the Dutch or from the Netherlands Top Sectors platform. That's about 2 million euros, which is basically focused for the development and implementation of key breeding technologies, which should be specific to our breeding program. So we really are trying to bring, you know, the highest end technology that you can have in breeding to this quinoa breeding program. And the idea is to just do things faster, but also with a lot more precision. I mean, where we want to get to is that we can release varieties every 3 years. So really, that's our ambition. And for that, of course, what you want to do is you want to be able to use molecular marker technologies, use the best phenotyping facilities, take advanced phenotyping technologies to the fields. And so I'm very excited today because I get to show you a bit of what we're doing here. And this is, of course, more the work of Robert. So I'm very happy to explain it to you. But of course, if you have more questions, we can always contact Robert to answer the more technical details about these tools. But the first thing that I should be talking about, of course, is that we are introducing this Allegro targeting genotyping system into our breeding program. And basically, this platform allows you to use the already published quinoa genome to develop customizable target S&P marker sets. And the technology is fantastically versatile, and it's also very cost effective. And basically what we have done as a proof of concept is that we have developed a set of 5,000 markers, and we have applied this to a new mapping population just to see how well it works. And the results are really nice. Actually, what you can see here in this graph is the new genetic map that we've created with our Allegro technology And you can compare it with the map that was created for the Quinoa genome paper, which, by the way, is also a population which has been developed by Radical Crops and Wageningen University. And basically, the conclusion was that all the elements of this new map fall perfectly with the elements of the Quinoa genome map that we've created. So basically, the technology works perfectly fine. And so now what we're actually doing is that we are using these markers to play with our mapping population. So we have several mapping populations which capture very well the diversity of the breeding program. And basically what we're trying to do now is look for QTLs for simple traits like the bitter, non-bitter. So it would be fantastic if we can just use molecular markers to tell whether something is saponin-free or not. But we're also looking at very complex traits like lodging or drought or salinity tolerance. And you can see here that we already have some examples of the results of these QTL mapping experiments. So we're really moving forward in that sense. The idea is, of course, like I said, because these mapping populations, they capture the diversity of our breeding program. We can directly apply these QTLs in our breeding program, so we can use them as selection markers. So that makes things very effective. And of course, the whole idea, the future for us is to go through genomic selection. Like I said, we want to apply speed breeding, so we really try to go for at least 3 cycles per year. And then we want to be able to select, without actually having to phenotype. So building genomic selection models is going to be a must for us. And this Allegro genotyping technology, because it's customizable and so scalable, it will allow us to do this in a cost-effective manner. Perfect. Now, the Allegro technology will also allow us to introduce new traits from novel germplasm, and we're also using it for genetic barcoding. So this is very important for our business. We don't want to have infringements of PVP. And so now we've created a set of SNP markers that can help us perfectly identify the varieties and also to trace if someone is illegally using them, which is very bad for our clients because they're actually paying the royalties to be able to produce quinoa with these varieties. So it's very important to have these tools. The other reason why this marker set is so important is because we are now working towards commercial hybrids by using cytoplasmic male sterility. So here you have a really cool example of what we are trying to achieve. So here you have a CMS mother. So this plant has a CMS background. It's not a nice looking plant, but we're making a lot of progress in actually introducing the CMS background into our Dutch variety. So you can see already that you have some very nice plants or better looking plants that are going more towards the elite material that we have. But they have this cytoplasmic male sterility. So this is really something amazing because, if you can create F1 quinoa hybrids, then you're maximizing the power of heterosis and you can probably even break yield ceilings in quinoa. But it's not only about producing this hybrid seed platform. It's also about testing combinations that can help you make the right hybrid. So this year we're actually also doing a D-allele test crossing experiment to test for combining and specific general ability in our elite lines and varieties. And definitely the marker system will be very important in facilitating these efforts. It's not only about markers, it's also about phenotyping. And one of the great things about working at Wageningen University is that we can access the Netherlands plant eco-phenotyping, just basically a bunch of amazing tools, greenhouses, drones, cameras, sensors that can help you really look deep into quinoa physiology. And the idea is that you can, for example, test complex traits, underpin the biochemical, but also the genetic mechanisms of traits like drought or salt tolerance or nitrogen use efficiency. And the idea is to be able to use this very advanced equipment to understand our germplasm, but also to create QTLs for selection. And the other thing that we're doing, of course, is that by having this NPEC facility available to us, we can also use very amazing multispectral imaging with drones. Basically, what we're doing is we're extending our testing facility. So we have 3 stations in the Netherlands, in the UK, and France. We're testing 500 families, variety trials. So it's a lot of work. And then you need some drones to help you. And these things are just amazing. They can let you predict yields. They can let you test production functions. They can even help you predict protein content, which is pretty amazing if you want to create genotypes that can yield more protein. So overall, pretty amazing stuff that I don't always understand. But the most important thing is that, you know, I think that we're making great strides towards making quinoa a global staple. As you can see from this presentation, the breeding program at Radical and Wageningen University has been scaled up enormously, especially compared to recent years. And so I'm very confident that we can take on the challenge of taking this wonderful crop and really harnessing its genetic potential. But again, we do this to support parties, especially agricultural firms that share our vision of a more sustainable, resilient, and biodiverse food system. We really want to turn quinoa into a global staple, and we want to support people who want to do this. So I just want to finish up by inviting you to visit our website, www.radicalcrops.com. You know, the idea is that you can get in contact with us. We're going to be more active in South America and North America in the coming year. And basically, I would like to invite you that if you want to start collaboration, a commercial collaboration in producing quinoa with our varieties, please visit the website, drop us an email. But also, if you just want to do research on this wonderful crop, also get in contact with us because we're very happy to continue to explore what makes quinoa so amazing. At the same time, I would like to take this opportunity to announce some opportunities at Radical. So we are really a young company, we're trying to grow. And one of the things that we want to have is a crop specialist in North America that can not only help us create more market for the varieties, but that can actually support existing clients. So if you have an affinity for quinoa agronomy, if you're passionate about plant breeding, please visit the website. I think you will find the job post there. And if not, you can just drop me an email at Andres, ANDRES, @radicalcrops.com. With that, once again, I really want to thank the organizers, but also the participants of the symposium for this amazing opportunity to share our story. Really, it has been fantastic to be here with you. Thank you very much. And of course, if you have any questions, please, I'm here to tell you as much as I can.
Thank you so much, Andres.
Muchísimas gracias, Andrés.
It was a beautiful talk, and we understandably have quite a few questions for you.
Fue una charla muy buena, muy bonita, y aquí tenemos algunas preguntas para usted.
Our first question is from Christina Ocana.
Nuestra primera pregunta viene de Cristina Ocaña.
Andres, have you found that saponin-free quinoas attract more pathogens?
Andrés, ¿has observado si las quinoas libres de saponina atraen más patógenos?
Would there be a trade-off between having to spend money on desaponification versus having to spend it on insecticides?
En ese caso, ¿se gastaría la misma cantidad de dinero en insecticidas que en la desaponificación?
So I can talk now, right? Or
Yes.
Sorry, I'm terrible with technology, but thank you, Christina, for that question.
Muchas gracias, Cristina, por la pregunta.
Just to keep it short, we haven't seen any negative correlation between you know, having saponins in your quinoa grain and having more resistance or less resistance towards pathogens. I think sometimes people tend to tell us that. Oh, should I stop in between? So that
sí, Andrés, por favor, para que me permitas traducir, si no se me olvida. Muy bien. Andrés dice que en realidad no han visto una correlación entre la saponificación y el hecho de que atraiga más patógenos o no.
Yeah, and you know, just to add something else, I mean, sometimes you see that in experimental plots, birds might like the saponin-free quinoa better than the one with saponins. But you know, this just happens at a small scale. On a large scale, we don't really see significant bird damage on the saponin-free varieties.
Y puede ser que en pequeños experimentos de laboratorio usted pueda ver alguna diferencia en la saponificación con respecto a los patógenos, pero cuando hablamos de cantidades grandes, en realidad no vemos ninguna. ¿Y cuál era la palabra, Andrés?
A los pájaros.
Ah, los pájaros, perdón.
Beautiful. Our next question is from Thomas Davis.
Nuestra siguiente pregunta es de Thomas Davis.
Thomas wants to know, is the Allegro technology published?
Thomas quiere saber si la tecnología Allegro ha sido publicada
Or available to the Quinoa community as a service?
o si está disponible para la comunidad de Quinoa como un servicio.
Yeah, so basically, Thomas, right? What I can tell you is that Allegro is actually a commercial product. So I can even add the information where you can find it.
Lo que le puedo decir es que Allegro es un producto comercial. Entonces yo le puedo compartir la información donde usted la puede encontrar.
There are several publications describing the use of the technology for example, for tomato. And currently our breeders are developing, let's say, or testing the technology in quinoa.
Okay. Hay muchas tecnologías, especialmente para tomate, por ejemplo, pero en estos momentos nuestros mejoradores de semilla están tratando de utilizarla para la quinoa.
And basically it will be published at some point, but basically we are at the validation phase. Now we're using it in the breeding program, and I think some already interesting results have come out of it. So I hope that we can soon share the technology itself and how it works in quinoa to the rest of the community. But yes, that's something that's in the horizon.
Bueno, y por ahora no está publicado todavía. Es algo que estamos buscando hacer en un futuro y por supuesto que va a ser muy, muy importante para nosotros. Andrés, que se me olvidó.
Estamos bien.
Beautiful. Our next question is from Carla Little.
Nuestra siguiente pregunta es de Carla Little.
Carla is coming from Bolivia.
Carla viene de Bolivia.
And she's wondering if you have explored digital imaging for phenotyping diseases.
Y ella se pregunta si ustedes han explorado imágenes de fenotipos para especies. Rachel, can you repeat that last part, please?
For phenotyping diseases.
Para enfermedades fenotípicas o de fenotipos.
Hi, Carla. Thanks for the question. Actually, as far as I'm aware, no, not yet. I mean, that would be really groundbreaking technology that we should apply to the breeding program. But currently, it's not in use. So if you have ideas of how to do it, or if you have some research interest, just drop me an email and then we can continue that discussion.
Carla, esa es una pregunta muy buena e interesante. En realidad no tenemos ninguna. Eso sería un campo muy interesante para estudiar en nuestro laboratorio. Así que la invito, si usted tiene algunas ideas o si tiene información, compártala conmigo por un correo electrónico y nos ponemos en contacto.
Thank you, Carla, for your question.
Gracias, Carla, por su pregunta.
The next question is from me.
La siguiente pregunta viene de Rachel.
How does your farmer agronomic support network vary by country?
¿Cómo es que su network agronómico está apoyado en cada país?
Who are the extension partners you build into your network?
¿Quiénes son sus colegas que ustedes agregan a su network?
That's a great question, Rachel. Yeah, so most of our partners they've developed the production chains themselves. And basically, they are the ones that have generated the knowledge about how to cultivate quinoa in the regions.
La mayoría de nuestros afiliados son los que han generado la tecnología ellos mismos y entonces son los que cultivan la quinoa en sus regiones.
And so basically, what we have done as a company is that we create platforms so that we can meet between all these experts to share ideas, to share thoughts on the machinery that you should use, experiences on how to do, mechanical cultivation of weeds, et cetera, et cetera. So that's how we're doing it currently because we are mostly present in Europe.
Ajá. Y entonces lo que hacemos básicamente en nuestra compañía es reunirnos con ellos y compartir las experiencias que cada uno tiene sobre cómo cultiva, sobre cómo manejan las enfermedades, sobre cómo manejan las malezas, sobre qué hacen en los diferentes estados o tiempos del cultivo, y entonces así nos mantenemos informados.
And what we're doing now is that the network itself, it's helping us when we go abroad. So if we have a new opportunity in the US, people from the network will help me set it up. And what we would like to have ideally is people from our company, like I said, based in the US that can help us, that can serve as these, knowledge extensions to clients in different parts of the world. And that needs to happen because, the world is pretty big and we're still a startup.
Bueno, y en realidad lo que eso hace es que nos ha ayudado a mantenernos en contacto con diferentes agricultores en diferentes partes del mundo. Entonces, cuando viajamos a un país diferente y necesitamos conexiones o necesitamos información, vamos hacia ellos y nos conectamos. Y esto es algo que necesitamos hacer más a menudo porque el mundo es muy grande y hay muchas personas cultivando y haciendo este trabajo. Entonces, entre más conectados nos mantengamos, mejor.
Thank you so much, Andres.
Muchas gracias, Andrés.
Our next question is from John Huilca Quisepe.
Nuestra siguiente pregunta es de John Wilka Quisepe.
Thank you for your presentation.
Muchas gracias por su presentación.
What prospects do these improved varieties have without saponin?
¿Cuál es el futuro de estas nuevas variedades sin saponificarlas?
In the Andes, South America, to climatic and pathological adversities?
En los Andes y en Sudamérica para las adversidades que tienen que ver con patógenos y con el medio ambiente.
Thanks, John, for the question. So we've already worked in several countries in the Andean region. We've worked in Bolivia and we've worked in Ecuador, and we've also worked in Chile. I think you can also consider Chile an Andean country. And, the varieties work very well over there. So what you see is that, of course, we've created short cycle varieties. So they have shorter cycles, and that helps you escape several stresses.
Okay. Bueno, pues le puedo decir que nosotros hemos visitado Bolivia, Ecuador, incluso Chile. Podemos considerar Chile como parte de los países andinos. Y lo que le puedo decir es que hemos visto que utilizan ciclos más cortos y esto ayuda con manejar un poco más todos estos factores adversos que se pueden presentar.
So if you ask me about the prospects, I can tell you that, there's a lot of interest, and I hope that we can start working with producers in South America who believe that there's an advantage to having saponin-free varieties.
Uh-huh. Pues si usted me pregunta por el futuro en esto, yo le puedo decir que hay mucho interés en Sudamérica por empezar a cultivar variedades que estén libres de saponificación.
Andres, thank you so much.
Andrés, muchísimas gracias.
We still have some questions for you that will be moving to Slack.
Tenemos otras preguntas para usted, pero las vamos a mover al canal de Slack.
So if anyone has additional questions for Andres, please go there.
Si alguien tiene preguntas o más preguntas para Andrés, por favor vaya a nuestro canal de Slack. Ahí pueden ver las respuestas, pero también pueden hacer más preguntas.
And Andres, I hope you can post your open position on Slack as well.
Y Andrés, yo espero que usted pueda publicar la posición o el trabajo que tienen allá abierto en nuestro canal de Slack también.
We have a channel specifically for job opportunities.
tenemos un canal específicamente para oportunidades de trabajo.
So thank you again, and I hope you can stay on to watch Anna's talk as well.
De modo que muchas gracias de nuevo y espero que usted se pueda quedar para ver la charla de Ana o la presentación de Ana que sigue a continuación.
With that, we'll introduce our next speaker, Anna Teston.
Con esto vamos a presentar a nuestra siguiente oradora, Ana Tester.
One moment, everyone. We'll figure out the audio situation momentarily.
Les pedimos por favor un momento a todos. Estamos tratando de solucionar los problemas de audio que tenemos en este momento.
It's my pleasure to introduce our speaker, Anna Teston. Anna is a plant pathologist with the USDA ARS Plant Introduction Research Unit in Ames, Iowa. She holds a PhD from the Ohio State University in plant pathology and a master's from as a virtual Hello, everyone. To those just joining us, this is the Horizons in Quinoa Production System session. My name is Rachel Breslauer, and I'm going to be your facilitator for this session. I am a PhD student working in the Sustainable Seed Systems Lab at Washington State University. It's my pleasure to introduce our speaker, Anna Teston. Anna is a plant pathologist with the USDA ARS Plant Introduction Research Unit in Ames, Iowa. She holds a PhD from the Ohio State University in plant pathology and a master's from the Pennsylvania State University in plant pathology and international agriculture and development. Anna's research and expertise focuses on disease detection and management in specialty crops. And she's worked in Ecuador, Bolivia, Tanzania, and Ethiopia. In her master's, she studied the use of plant growth promoting bacteria to promote quinoa growth and developed a method to detect quinoa downy mildew pathogen in the seed. I'm very excited for her talk, and I'd like to welcome Anna Teston.
All right, well, thank you so much, Rachel, for that introduction. As a plant pathologist, I'm really passionate about plant diseases, and I know a lot of times you don't like seeing a plant pathologist come to your field because we will find problems. But in my talk today, I really want to go over some basic information on what the most prevalent diseases are in quinoa, but I also want to go over where we need to go as a research community so that we can address these global challenges to quinoa health. So I first started working with quinoa in my master's at Penn State, and in my current role as plant pathologist in the Plant Introduction Research Unit, I have access to the USDA's collection of quinoa germplasm. So I really like to think about this question of, as quinoa production expands to new regions globally, how do we address existing and emerging quinoa diseases? because as we introduce crops to new environments, new challenges in terms of plant diseases will always arise. So in my talk today, I'm going to go over major and minor or emerging diseases of quinoa. I'll address some major gaps in our knowledge of these diseases, and I'll focus mostly on quinoa downy mildew. And then I'll go over how we can address these gaps and challenges as a research community. So in addressing this question, I like to introduce the concept of the disease triangle. Now, if you've ever taken a plant pathology course, I'm sure you've seen this triangle. And this is just a way to conceptualize plant diseases. In order for a plant disease to occur, we need a virulent pathogen, a conducive environment, and a susceptible host to be present. But again, if we think about quinoa production expanding globally, the pathogens, environments, and host genetic backgrounds that we will be encountering in these new production systems will all be different. So we're going to have new diseases that are emerging and also existing diseases that become a bigger problem. The other thing I want you to keep in mind in this talk is that quinoa diseases do not occur one at a time. And this is true for most crops, but it makes it very difficult from a disease management standpoint to deal with multiple pathogens at once. Now, this is a picture of some quinoa leaves from a trial in Pennsylvania. And what's really interesting about these leaves is that they're infected with, they have downy mildew, they have ascochyta leaf spot, and they have passiflora or cercospora leaf spot. So these poor plants are being hammered by 3 different pathogens at the same time. Now, why this is important to think about in the global context with quinoa is that as we're introducing quinoa to new environments, the interactions of these pathogens have the potential to change, and we can see new diseases emerging in these new contexts. So let's jump right in to see what some of the most major quinoa diseases are. And I'm going to start with the foliar diseases of quinoa that are caused by fungi. The 2 most commonly fungal leaf spots of quinoa include ascochyta leaf spot and passiflora or cercospora leaf spot. Now, ascochyta leaf spot has been described as being caused by both Ascochyta callina and Ascochyta hyalospora. And the actual species distribution that causes this disease is a bit up in the air, and that's something we need to do more research on. For passiflora or cercospora leaf spot, this disease is known as ojo de gallo in South America. This is caused by Passiflora dubia, which is formerly known as Cercospora dubia. But it's really likely that given quinoa's really broad growing range, that there are other Cercospora species that are causing these foliar leaf spots. So you'll hear me say multiple times in this talk that we need to develop a better understanding of what pathogens are actually causing these diseases. Now, another foliar disease of quinoa that's caused by a fungus is mojo verde or green mold. This disease I've only seen described in South America, and it's caused by Cladosporium species. Now, Cladosporium as a plant pathogen is a weaker plant pathogen. It usually attacks senescent or older tissues on the plant. And this pathogen has been described as attacking both leaves, but also the panicle of quinoa. Now, because we haven't seen this disease outside of South America, we really don't know how much of an impact this is going to have on quinoa production globally. Now, one type of diseases of quinoa that I think will be very important as quinoa is introduced to new growing ranges are the stem diseases of quinoa. These have potential to cause tremendous yield loss if they cause lodging in the plant before harvest. So Phoma stock rot or brown rot is caused by several species of Phoma. This has been described in South America, and it's also been described in Canada in quinoa production. Now, again, as I said, with the foliar leaf spots, we're really not sure what species of Phoma are causing this disease across these growing ranges. So we need to conduct more research in this area. The next disease is Ascochyta stem rot, and this is interesting because this is an emerging disease in quinoa. It's also been described as quinoa black stem. This disease is caused by Ascochyta callina, which, if you remember from our foliar diseases, also causes Ascochyta leaf spot. Now, when I worked with quinoa in And there's a very good publication from a research group in China describing quinoa black stem and its management, as this is an emerging disease in Chinese quinoa production. Now, again, I said that diseases will emerge when quinoa is grown in new areas. And in 2019, I saw this happen in Iowa at my own research station. And in 2019, we saw new stem lesions developing on a quinoa demonstration planting that we were growing. You can see these silvery to black lesions on the stem. And when we isolated fungi from these, we found that it was both Colototricum nigrum and Colototricum truncatum. And these are causal agents of anthracnose. Now, I dug through the literature as best as I can, but I can't find any report of Colototricum infecting quinoa anywhere in the world. So this is a novel disease. Moving on to root and crown diseases of quinoa. In terms of damping off is a perennial problem in many crops. This really can limit yield if it prevents you from having a good stand. In quinoa, the species of pathogens that have been described as causing damping off include Rhizoctonia and Sclerotium, which are both fungi, and Pythium, which is an oomycete. Fusarium, which is a fungal pathogen important in a lot of different crops, also causes damping off in quinoa, but it also causes root rot and wilt. If you look at this picture here, you can see that the inside of this root is pink, and that's very characteristic of Fusarium infected quinoa roots. This is a picture from a collaborator, Giovanna Plata, at Quinoa Foundation in Bolivia. And these are quinoa plants that have Fusarium root rot. And you can see that those root systems have been absolutely destroyed. So given the right environmental conditions and fields with a history of Fusarium, this could be a tremendous yield limiting biotic factor in quinoa production. Most of the research on nematodes in quinoa production has been performed in South America. So Globodera and Nacabis have been reported, and I'm not going to say this last name because it's a Friday and it's too long of a Latin name to say. But I do want to make the point for nematodes. The distribution of nematodes is very dependent on a cropping history in a region and also the environmental condition in a region. So as quinoa is introduced to new regions, it's likely that we'll see new nematode pests of quinoa emerging. And I want to take a moment to wrap up with something that's really interesting to me, and that's the relationship of bacteria and viruses to quinoa. So bacteria and viral diseases in quinoa are not very well classified, but we know they have to occur. In terms of bacterial diseases, there have been few recent or definitive reports of bacterial diseases in quinoa. I found a publication from 1975 describing a bacterial disease in quinoa. The author was able to identify this as a Pseudomonas disease. More recently, Quinoa Foundation has identified a Pseudomonas species that causes a leaf spot and a stem rot, but we really need to work harder to classify this Pseudomonas down to species and identify if we have multiple species causing these diseases. In terms of quinoa viruses, this is a really interesting plant pathology story. So quinoa in the field of plant pathology is used as a bioindicator plant for viral infections. Quinoa is commonly used as a diagnostic tool. So if you're going to try this, if you have a plant, say a cucumber, that you think has a virus, you would grind up leaves from that plant and then rub that suspension on quinoa leaves. If there's a virus present, you're going to see these yellow spots on the quinoa leaf, and those are called local lesions, which indicate that the quinoa plant is fighting a virus. Now, this is really cool as a plant pathology tool, but it makes searching the literature for quinoa viruses very difficult, because more often than not, if you look for quinoa and a specific virus, all you can find is quinoa being used as a bioindicator plant. So we really don't have an idea of what viruses commonly occur in quinoa in nature. And so this is really an area where further characterization is needed, not just in quinoa's native Andean range, but also in introduced regions. And I know there's some work that's being started at Washington State University to look at what viruses are occurring in quinoa. So in the last part of my talk, I'd like to move on to my favorite quinoa pathosystem, and that's the quinoa downy mildew pathosystem. And this is a picture of quinoa that I saw in Bolivia in a field outside of Cochabamba. And if you look at this picture, you can see that nearly all of the plants have yellowing leaves, and all of those yellow leaves are infected with quinoa downy mildew. Now, from a plant pathology standpoint, to have 100% incidence of a disease in a field is very devastating. And quinoa downy mildew is the most devastating disease of quinoa. Because it's the most devastating disease of quinoa, luckily we know a bit more about this disease than we know about other quinoa diseases. So quinoa downy mildew is caused by Peronospora variabilis. It was formerly known as Peronospora farinosa, forma specialis chenopodii. This, as I said, is the most important quinoa disease globally. And the reason for this is that it's a seed-borne disease. In this picture at the bottom, these orange structures are called oospores. Those are the survival structures of the quinoa downy mildew pathogen. And unfortunately, those oospores can adhere to seeds and spread wherever that seed is introduced. So this really has facilitated the global spread of quinoa downy mildew. This disease, as I said, can cause severe yield losses, and given the right environmental conditions, it can lead to complete yield losses. From a plant pathology standpoint, quinoa downy mildew is an obligate heterophthalic oomycete pathogen. Now that is a lot of plant pathology jargon, but I want to break it down for you. What I mean by obligate is that the quinoa downy mildew pathogen requires a living plant host for survival. If there's not a living quinoa plant in the field, that pathogen can just sit there as an oospore and not do anything. It needs to parasitize a live plant. Quinoa downy mildew, Peronospora variabilis, is also heterophthalic, which means there are 2 mating types that are required for oospore formation. Why this is important is that if you only have 1 mating type present, that can limit the genetic diversity of the pathogen population in your field, which can hopefully make management easier. And it's an oomycete pathogen, which means it's a fungal-like organism that's more closely related to plants than the true fungi. And so we need to keep this in mind when we come up with management strategies for this. But the fact that this is an obligate pathogen and also heterophthalic makes Peronospora variabilis very difficult to study. So many of our other plant pathogens we can grow in the lab on artificial media, but for the quinoa downy mildew pathogen, we need to grow that along with living plants. So it makes it very difficult to conduct experiments with this pathogen. Now, because it's very difficult to study, there's a lot of research areas that we need to explore to help us ultimately come up with better management strategies for this disease. Now, one of the very important research questions we need to answer is how many Peronospora species actually cause downy mildew? You might think that's a strange question because I said it's just caused by Peronospora variabilis, but Peronospora variabilis appears to be the dominant species causing quinoa downy mildew, but we know there are other species. So this is a picture of a quinoa plant taken in the Chimborazo province of Ecuador. And you can see that these leaves have some light green discoloration and a cupping of leaves. And that's really not a typical symptom of quinoa downy mildew. And my collaborator, Jose Ochoa with INEAP, had seen this symptom in this province for quite a while and was curious as to whether or not it was a novel Peronospora or it was just a weird genetic interaction. And in my phytopathology paper from 2014, we sequenced 2 genes and found that based on phylogenies of both of those genes, this is a novel species of Peronospora capable of causing disease in quinoa. Now, this was just in one region of Ecuador, but again, quinoa has a really broad growing range, and there's potential that there are other species of Peronospora that are causing downy mildew. Now, to take this a step further and make it a little more complicated, within each of these Peronospora species, we have different races. What I mean by race is that this is a specific strain of the pathogen that can only infect specific genetic backgrounds. And knowing race structures of pathogens is very important in the breeding of disease-resistant crops. But for Peronospora variabilis, we know based on work, again, of Jose Ochoa, that there is a race structure. And Jose identified, I believe, 4 to 5 different races. But that's likely just the tip of the iceberg. If we look at the most closely related other downy mildew pathosystem, which is the spinach downy mildew pathosystem, that's caused by Peronospora effusa. There are 17 different races of Peronospora effusa. So there is a lot more we need to learn about Peronospora variabilis in order to start breeding resistance to this strain, to this pathogen. Another question we need to answer about Peronospora variabilis is, what is the host range of this pathogen? And this is really important in terms of basic disease management. So we want to answer this question of, what role do weedy Chenopodium play in disease outbreaks? And we know, based on recent publications out of Turkey, that Peronospora variabilis is capable of infecting Chenopodium album in that country. When I worked with Peronospora variabilis, I was never able to get it to infect weedy Chenopodium native to Pennsylvania, but I was able to get my strain of Peronospora variabilis to infect Quinoa, or Chenopodium paludicale, which is another Andean edible chenopode. So from a management standpoint, if weedy Chenopodium are important reservoirs of this pathogen, we really need to focus management on managing these weeds, not just in quinoa fields, but surrounding quinoa fields. And there is an example from the Passiflora leaf spot that weedy Chenopodium are an important reservoir of this pathogen, at least in North America. So ultimately, again, we want to be able to better manage quinoa downy mildew. The foundation of any disease management program really needs to rest on the use of resistant varieties. When I worked with INEAP and ProIMPA Foundation, there were breeders working to develop quinoa downy mildew resistant varieties, which is great. In terms of having tools for organic production of quinoa, biological control is an important tool. And ProIMPA Foundation in Bolivia has really pioneered the use of biocontrol in indigenous Andean crops, using indigenous bacillus and trichoderma strains isolated from those crops. We need better tools to both monitor and predict these diseases, and I'm going to talk about that a little more shortly. And last but not least, we need to work on identifying what fungicides are most effective, and also developing ways to reduce the development of fungicide resistance. So I want to take a minute now to talk about what are some very broad needs in terms of quinoa disease research so that we can improve disease management of quinoa globally. First, we need easier and improved access to information on quinoa diseases. A lot of times you have to dig through obscure journals to find information of a report of a disease that was reported just once. And so I encourage researchers working with quinoa diseases to make official reports of new diseases in an international journal such as Plant Disease, or new disease reports that's put out by the British Society for Plant Pathology. We need to work on having naming standards for the diseases and pathogens that we're working with. If you remember from the fungal pathogens of quinoa, a lot of these have not been properly identified to species. And the first step of disease management is identifying that pathogen properly. And so we need to use modern molecular tools such as sequencing to identify what species are actually causing these diseases. Second, we need to work on having standardized disease names for quinoa diseases. Oftentimes, I've seen publications of a quinoa being grown in a new region, and they'll give a disease name that's completely different from a name that was given in South America. So I recommend that people look at Plagas and Enfermedades del Cultivo de Quinoa, which was put out by ProIMPA Foundation. And this is a very good overview of the disease names that are used in quinoa in South America. Next, we need to come up with standardized methods for working with these pathogens and diseases. Solveig Danielson and Therese Ames put together an excellent guide for working with quinoa downy mildew. This is freely available on the internet. And if you're starting to work with downy mildew, I really encourage you to check this out. Last, I would like to encourage collaborative efforts on quinoa disease management as quinoa is introduced to new countries. So there have been researchers in South America who've been working with these diseases for years, and it'd be really good if we could establish linkages between these researchers and researchers in new regions so that these researchers in new regions don't have to reinvent the wheel. All right, what are some overall research needs to improve disease management? First, and this is true of all crops, we need to improve our understanding of the epidemiology of the diseases we're working with. In quinoa, again, the most important disease is downy mildew. We need better tools to diagnose, monitor, and predict these diseases in the field. In terms of diagnostic tools, simple PCR specific primers and qPCR primers are excellent tools for detecting these pathogens. In terms of monitoring tools, using tools such as spore traps or early detection using drones are also potential ways to improve disease management. And then in terms of disease prediction tools, developing models based on climatic data can really help farmers know when they need to apply disease management practices. I was really happy to see that there are modern genomics tools being applied to quinoa breeding. These genomics tools can also be applied to the pathogen side in order to develop a better understanding of the pathogen populations and also to develop better diagnostic tools. I'd encourage anybody working with downy mildew to look at the literature for spinach and cucurbit downy mildew research, because these are very advanced fields that have developed tools that can be readily used by growers. And I think these tools could be pretty easily adapted to quinoa downy mildew management. Next, we need tools that will work across quinoa production systems. As quinoa is becoming quickly a global crop, we need tools that will work not just for smallholder farmers, but also to large industrial agriculture production. So unfortunately, quinoa diseases will be an issue wherever quinoa is produced. Thankfully, there's plant pathologists who are always ready to work with these diseases. I like to use the example of another famous Andean crop, potato, and its most important disease, late blight. Wherever potato has been introduced in the world, late blight has always been a problem. So we need to be prepared for quinoa that downy mildew will always be a problem wherever we grow this crop. But we can create tools to improve detection and management of these diseases. And by working collaboratively, we can improve quinoa health worldwide. And with that, I'd like to thank the organizers of this research symposium. And I'd like to also thank the funding sources for my research during my master's, which was USAID, and especially my collaborators in Bolivia at ProIMPA Foundation and my collaborators at Ecuador with INEAP. Thank you.
Thank you, Anna, for your talk.
Muchísimas gracias, Ana, por su presentación.
It's beautiful, it's broadly applicable, and I think this will be useful for people who are not plant pathologists as well as people who love plant diseases.
Es una presentación muy bonita, aplicable, no solo para la gente que está en el ámbito de las plantas y la patología de las plantas, sino también para las personas comunes y corrientes para aprender un poco más sobre todo esto.
Our first question comes from Julianne Kellogg.
Nuestra primera pregunta viene de Julian Kellogg.
What are effective organic or conventional seed treatments?
¿Cuáles son algunos tratamientos convencionales o orgánicos para las semillas?
To prevent distribution of seed carrying downy mildew.
Para prevenir la distribución de semillas que tienen mildew.
That's a great question, and it is something that hasn't really been addressed in the literature, but we can make some guesses as to what will work based on other seed treatments. So for,
esa es una muy buena pregunta, y es algo que en realidad no está en la literatura, no se puede encontrar en la literatura todavía, pero es algo de lo que podemos hablar y más o menos adivinar, si se quisiera decir un poco, qué es lo que podemos hacer.
So for organic production, likely because quinoa seeds are somewhat delicate, we can look at steam or dry heat seed treatments to eliminate the downy mildew.
Para las producciones orgánicas, como nosotros sabemos, la quinoa es una semilla un poco delicada. Entonces podríamos mirar a tratamientos que tienen que ver con la germinación de la semilla. To reduce what, Ana?
To reduce the downy mildew.
Sí, para reducir el mildew.
And for conventional production, so steam and dry heat would also work in conventional. But for conventional, we also could use fungicides such as metalaxyl.
Y para las producciones convencionales o la quinoa convencional, también se podría utilizar el calor y la sequedad, que fue lo que dijo anteriormente, pero en este caso también podemos utilizar químicos para tratar, o los fungicidas para tratar esta enfermedad.
Excellent. The second question comes from me.
La siguiente pregunta viene de parte de Rachel.
How can non-plant pathologists at our symposium support the research needs identified in your talk?
¿Cómo es que las personas que no están en el área de patología de plantas pueden apoyar la investigación que ustedes llevan a cabo?
That's another great question. So thanks for that.
Eso es una muy buena pregunta. Gracias por eso.
So I think the easiest way is to just reach out to somebody that has worked with quinoa diseases previously.
Y yo creo que la respuesta más fácil es decirles que lo primero que tienen que hacer es ponerse en contacto con alguien que tenga experiencia con el tratamiento de enfermedades de la quinoa.
The body of literature on quinoa diseases is very small, so there are very few people who have trained in this, but that expertise really needs to be utilized, especially with these new genomics tools and in breeding of disease-resistant lines.
La verdad es que la literatura referente a las enfermedades de la quinoa es muy pequeña. Entonces tenemos expertos en el tema y por eso es importante que se pongan en contacto con un experto para así poder tener un mejor resultado en el mejoramiento de semillas.
And a follow-up to that, are there any resources that could come out of the symposium that we could use to connect these two, to connect these two nodes?
Ok, y dentro de ese mismo tema, ¿hay algunos recursos que puedan salir aquí de ese simposio donde se puedan conectar o que puedan conectar estos dos temas?
I think one thing we could do following the symposium is maybe make a small working group of people who have presented on diseases and link those with other researchers we know so that we have a broader community that can reach out.
Bueno, yo creo que una de las cosas que podemos hacer es que al terminar el simposio, creemos un grupo en el que tengamos a todas las personas que han presentado sobre enfermedades y de esa manera podemos hacer un grupo, un network de contactos donde podemos continuar hablando sobre el tema y nos estaremos comunicando.
That's a great idea, Anna.
Es una muy buena idea, Ana.
We have another question for you from Carla Little.
Y tenemos otra pregunta para ti de Carla Little.
For downy mildew scoring, how would untrained people recognize it out of other diseases?
Para la cantidad de mildew, ¿cómo es que una persona no entrenada podría reconocerla en medio de otras enfermedades?
Thanks, Carla. I hope you're doing well. So for downy mildew scoring, looking at that guide put out by Solveig Danielson and Therese Ames, there's some good pictures in there.
Para cómo ver la cantidad de mildew, mostramos unas fotos en la presentación. From who were the pictures, Ana?
Solveig Danielson.
Y entonces ahí puede ver más o menos cómo es que se identifican las enfermedades. Por medio de las fotos le va a dar una muy buena idea de cómo identificar esta enfermedad.
But in terms of developing new tools, I think myself and other quinoa pathologists could put together some photographic guides that would really help people in rating foliar diseases.
Pero en términos de otras herramientas que pueden ser útiles, yo creo que el equipo, unos colegas y yo nos podemos poner en la tarea de seleccionar algunas fotos que les van a ayudar en realidad a identificar las diferentes enfermedades en la planta de la quinoa.
Thanks, Anna. And everyone who's asking questions will be connected with Anna on Slack.
Muchas gracias, Ana. Y todos los que están haciendo preguntas, los conectaremos más tarde con Ana en nuestro canal de Slack.
So you can follow up and get some of the resources that she mentioned in her talk.
Para que ustedes puedan obtener algunos de los recursos que ella mencionó en su charla.
We're going to finish with one more question.
Vamos a terminar con una pregunta más.
This is also from Carla Little.
Esa también es de Carla Little.
Do you have any idea how many pathogens have been identified on quinoa, molecularly maybe?
¿Usted tiene una idea más o menos de cuántos patógenos moleculares o molecularmente se han identificado en la quinoa?
Another good question. Most of the work has been based on morphology.
Muy buena pregunta. La mayoría del trabajo se ha basado en la morfología.
Right now, the only molecular tool that I know of that can be used for diagnostics of quinoa diseases are the PCR primers that I designed in the 2014 phytopathology paper.
Ok, en realidad la única herramienta que yo conozco que se ha desarrollado para la detección de enfermedades en la quinoa es una que yo desarrollé en el laboratorio en el 2014 y que está en un ensayo que publiqué en ese mismo año.
And I think this really highlights the need to develop some simple PCR assays to detect more of these pathogens.
Y lo que esto nos dice es que necesitamos desarrollar más herramientas simples de PCR para poder identificar estos patógenos.
And just one more thing here. In terms of identifying the species we're working with, we do need to apply modern sequencing tools so that we know exactly what species we're working with because they really can differ across the world.
Okay. Y en términos de trabajar con las diferentes especies, en realidad necesitamos tecnología un poco más moderna para secuenciar estas diferentes especies, porque como ustedes saben, tenemos muchísimas diferentes especies alrededor del mundo.
Thank you so much, Anna. We actually have time for one more question that came up just now that I think is really interesting.
Okay, muchas gracias, Ana. Pues creo que tenemos tiempo para una pregunta más que acaba de entrar.
Junja Zhang asks,
Ginger Han ask, o pregunta,
do you have any suggestions for cultural practices that can reduce downy mildew in many types of places?
¿Usted tiene sugerencias para prácticas culturales que puedan reducir el mildew?
So that's a good question. Downy mildew is very difficult to control.
Es una pregunta muy buena. Mildew es muy difícil de controlar.
The key to controlling a disease like downy mildew is to control moisture on the leaves.
Una de las claves para controlar la enfermedad mildew en las plantas es en realidad controlar la humedad de las hojas.
So in terms of cultural practices, anything you can do to reduce leaf moisture, such as adequate plant spacing, alignment of your rows, timing your planting so that you can maybe skip very moist periods of the year.
Bueno, y en términos de plantar, si usted puede plantar las plantas separadas, si puede hacer buenas hileras, si puede plantar en un tiempo del año que no sea tan húmedo, todos los factores que ayuden a controlar la humedad
And ultimately, we do want to have disease-resistant lines. That's going to be the foundation of any good downy mildew management program, but there's still a long way to go before those can be widely deployed.
Y en realidad lo que quisiéramos tener es resistencia en las plantas a esta enfermedad de mildew. Y al final del día, eso es lo que buscamos siempre. Una buena semilla que tenga resistencia a esta enfermedad sería fantástico, pero todavía tenemos mucho trabajo por hacer y mucha investigación por hacer.
Thank you so much for that advice, Anna.
Muchas gracias por su consejo, Ana.
This is an update from the Symposium planning team.
Esta es una actualización del equipo de planeación del simposio.
We are creating a channel in Slack specifically for pathogens and diseases.
Estamos creando un canal en Slack específicamente para hablar de patógenos.
So people who are experiencing pathogens in their field and those who have expertise to offer can come together.
Okay. De esta forma, las personas que tienen problemas con patógenos en sus cultivos o los expertos que saben sobre patógenos pueden unirse y conversar.
So thank you all for your questions. And
Muchísimas gracias a todos por sus preguntas.
thank you to our speakers for sharing so much knowledge with us today.
Muchísimas gracias a los presentadores por compartir tanto conocimiento con nosotros.
With that, I will be handing it over to Evan Crane for some final comments.
Y de esta manera dejo aquí encargado a Evan Crane para sus últimas palabras sobre esta sesión.
Thank you, Rachel. And I'd just like to once again thank the speakers and also Rachel for doing a wonderful job facilitating this session.
Muchísimas gracias, Rachel. Y yo también quiero aprovechar para agradecerle a los presentadores por su tiempo, por su presentación y para agradecerle a Rachel por todo su trabajo durante esta presentación y durante el simposio.
So please visit the Slack channel to follow up on any conversations started during this session.
Por favor, visite nuestro canal de Slack para que se actualicen sobre las conversaciones que tenemos sobre las diferentes sesiones.
And coming up in room 2, we will have the participatory quinoa breeding session.
Y en la sala número 2, enseguida tendremos mejoramiento participativo de la quinoa.
Thank you. We'll be taking a short break until the next session begins.
Muy bien, vamos a tomar un pequeño descanso mientras nuestra siguiente sesión continúa. Muchas gracias.
We'll begin at 11:40, so about 7 minutes.
Empezamos a las 11:40, hora de Pullman, así que en 7 minutos más o menos para cualquier horario que ustedes tengan.
Hi, Jared. Hi, Jinda. Can you all hear me?
Hi, yes, I can.
Oh, great. Perfect. Checking audio here. How about JJ? I see Laura.
Hey, Julianne.
Hi, Julianne.
Hey, awesome.
And do you know, will Blake be joining us as well?
This is Jared, I've been texting with him and talking to him in the morning, and I'm hoping he will. I haven't gotten a final response yet or not, but I told him, when our video and Q&A would be happening.
Great.
He said he was going to try and make it, but he's got a busy day today.
Awesome.
So Julianne, oh, go ahead.
Go ahead.
I was just going to ask a quick logistical question. I can ask it on the chat as well, but how do you want me all to work with Slack and the questions? It seems like you have a good rhythm going in terms of moving them around, and I don't know if I need to be doing any of that or if between, yeah.
I think you should select how you want to do it. What's nice about keeping Q&A open is that we actually see people upvoting.
Okay.
However, if questions get moved to Slack, it goes over with both the English and the Spanish translation.
Okay.
So let us know which one you would prefer, and then we'll just let the team know in our technical channel.
It's easier for me just to be able to look at the Q&A right on the screen.
Okay, great. Yeah, so I'll let everyone know to keep the questions in there and that you'll manage them.
Great.
Hi, Julianne. I have trouble to get into Slack, and can I get question just directly from you verbally?
Yeah. So what's your issue with getting into Slack? I thought I saw you posting there. Maybe I didn't.
I post my question in Q&A.
Oh, in Q&A. Okay.
Yeah.
What is your issue with signing in?
I don't have account. I forgot my Gmail account, so I have to set up a new account probably.
Okay, yeah, try setting up an account and then just send me a text message or an email, and then we can troubleshoot after this session.
All right.
Julianne, do you need me to help with some of the translations of the questions in Slack?
What I think would be really helpful is if you could translate the questions in the Q&A, and in that case, we would actually have you move to an attendee role so that the questions are commented on rather than answered.
Okay. That's fine. I can do that.
I can move Shirley to be an attendee now.
Fantastic.
Actually, Amy, you will probably have to do that since she is a co-host right now.
All right, yeah, I think we are probably good to go. And we will just communicate with Amy in our tech messaging.
Okay. Can you hear me? Oh, yes. All right.
Evan, do you want to get started with any of the logistics for the room?
Yeah, we'll go ahead and get started now.
Okay. Hola a todos, vamos a empezar.
Welcome to the second session in room 2 on the third day of the symposium.
Bienvenidos a la segunda sesión en la sala número 2 en el tercer día del simposio.
As participants, you do not have the ability to share video or sound, so please use the Q&A box for asking questions during the presentation.
Recuerden que como participantes ustedes no van a poder compartir video o sonido, pero tienen la caja de preguntas o respuestas para que puedan hacer preguntas.
And please vote for questions that you would like answered. We will try to have the speakers address these questions during the time that we have for the questions.
Y por favor, lean las preguntas y voten por las preguntas que a ustedes les gustaría que se respondieran. De esa forma, nosotros se las haremos a los presentadores para que sean respondidas.
And for any questions that aren't answered either because we don't have time or because the speaker isn't able to join us,
y para cualquier pregunta que no pueda ser respondida, ya sea porque no tenemos tiempo o porque el presentador no puede estar con nosotros,
we will move all the additional questions to the Slack channels.
Vamos a mover todas esas preguntas al canal de Slack.
And with that, I'm going to turn things over to the facilitator for this session, Laura Lewis.
Y de esta forma le daré el mando a nuestra facilitadora de esta sesión, Laura Lewis.
Thank you, Evan, and welcome everyone to the participatory quinoa breeding panel.
Muchas gracias a Evan y bienvenidos a todos al mejoramiento participativo de la quinoa.
I'd like to welcome our speakers, Julianne Kellogg, Jared Zeistro, Junda Zhang, and Blake Richard.
Quiero darle la bienvenida a nuestros presentadores, Julian Kellogg, Jared Sistro, Janda Yan, y Blake Richard.
Participatory plant breeding results in regionally adapted cultivars well received by participating farmers and their communities.
El fitomejoramiento participativo consiste en crear cultivos adaptados a las condiciones locales y regionales, y al ser un esfuerzo comunitario son bien recibidos por los agricultores participantes y sus comunidades.
We will learn about the unique methods researchers and farmers have used to develop new quinoa varieties in the northwestern United States using participatory plant breeding methods.
Vamos a aprender sobre los métodos únicos que los investigadores y agricultores han utilizado para desarrollar nuevas variedades de quinoa en el noreste de los Estados Unidos, utilizando métodos participativos de fitomejoramiento.
Jared Zeistro of Organic Seed Alliance describes his participatory quinoa breeding project in Northern California.
Jared Zeistro de Organic Seed Alliance describe su proyecto participativo de mejoramiento de quinoa en el norte de California, Estados Unidos.
Junda Zhang of Lundberg Family Farms tells the story of developing red and black seeded quinoa varieties suited to the Northern California climate and global market.
Junda Zhang de Lundberg Family Farms cuenta la historia del desarrollo de variedades de quinoa con semillas rojas y negras adecuadas para el clima del norte de California y el mercado global.
Blake Richards of Wild Rose Farm developed tricolor quinoa populations specifically adapted to his Northern California region.
Blake Richards de Wild Rose Farm hablará sobre el desarrollo de poblaciones de quinoa tricolor adaptadas específicamente a su región en el norte de California.
And Julianne Kellogg of Washington State University describes a participatory quinoa breeding project that involved multiple farmers and farms in Western Washington.
Y Julianne Kellogg, de la Universidad Estatal de Washington, describirá un proyecto participativo de mejoramiento de quinoa que involucró a múltiples granjeros, agricultores y granjas en el oeste de Washington, en Estados Unidos.
Please refer to their full bios that you can find on the Quinoa Symposium website.
Los invitamos a que vean sus biografías completas en nuestra página web del simposio.
We'll now get started with our first talk.
Vamos a empezar con nuestra primera presentación.
Presentation titled, thank you for attending this virtual presentation titled Evolutionary Participatory Quinoa Breeding for Organic Agroecosystems in the U.S. Pacific Northwest. I'll be presenting on research that was conducted in 2015 and 2016 as part of my master's degree. It was by far the most fun, interactive research project I've been involved with. I encourage others to experiment with participatory research. This project was designed to develop heterogeneous populations specifically adapted to organic farms in the U.S. Pacific Northwest, and to develop specifically adapted uniform varieties using seed from plants selected by farmers. We designed our field experiments to test two hypotheses. One being participatory selection will improve populations, and two, selections made from populations will outperform commonly used varieties. So to achieve our objectives, we created this breeding scheme. And to test our hypotheses, we built the breeding scheme to form two trials. It all begins in 2012 when lab member Adam Peterson crossed and then grew out the F1 populations. That was then followed by growouts with no active selection. And then for this research project, we decided to apply the different breeding methods that are commonly used by farmers or by researchers breeding for organic systems. And in 2015 was the first year of this project. Before I really dive into the breeding scheme and trial designs, I first need to explain the two breeding methods. One of the breeding methods is evolutionary breeding. It follows a rather simple and clear progression, kicking off with creation of genetic diversity, so like making the crosses, multiplication of seeds from each cross, repeated sowing and harvesting of the population in one or more agronomic environments without active selection of individual plants, so it's just natural selection. And then you have the use of the seed in culturally relevant settings as food or feed or in further breeding. Evolutionary participatory breeding is our other method. The addition of participatory kind of clues you in as to what's going on. Farmers are involved in the breeding process. Trials are conducted on farmers' fields and often managed by farmers. Data is generated by breeders, and that provides the basis for the selection of the successful varieties. Farmer involvement in participatory breeding can involve setting breeding goals, making initial crosses, selecting among diverse progeny, evaluating experimental varieties, distributing seed. And in our project, farmers were involved in the setting of breeding goals and the selecting among diverse progeny. So the breeding methods trial will bring you back to the breeding scheme here. Because we wanted to test the effect of farmer participation, we used the breeding methods trial. In 2015, the first year of the project, we had 6 populations that were planted as a split plot, randomized complete block design. And we had farmers who all sat down and completed a survey about preferred quinoa traits. And these farmers and then researchers walked the plots, first flagging plants that they preferred with their individualized flagging tape, and then remove the plants that they believed were not desirable. The evolutionary breeding plots were not touched. So designed as a split plot randomized complete block design, the population was nested within breeding method, which was nested within replication. There were 3 replicates and 3 locations each year. However, we experienced trial failures caused by high temperatures during flowering, stem borer devastation, and pre-harvest sprouting caused by early fall rains. Therefore, we ended up with only 1 location per year. The hedgerow trial, very exciting news. There were originally 213 plants in total selected by farmers and researchers, but some selections lacked enough seed to plant the following season. There were 92 entries the final year and had 3 successful locations. All were on certified organic farms on the Olympic Peninsula in Washington, in Western Washington. The hedgerow trial, designed to determine whether the selections made from populations will, if they will, outperform commonly used varieties. So it was designed to be an augmented, randomized complete block design. The entries were not repeated checks, 3 check varieties. Although all 3 locations for this trial were in the same region, they were rather precipitation. So Chimicum, about 700 millimeters of annual precipitation, Quilcene, almost 1,400 millimeters of annual precipitation, and Squim, about 400 millimeters of annual precipitation. So that was encouraging for us to have distinct environments for these trials. So the results for the breeding methods trial, this is the raw yield data. For the analysis, the data had to be transformed depending on the year. The mean of 3 replicates and standard error is reported. The parents of the populations are highlighted in green here. We expected the evolutionary breeding populations to have higher yield than the evolutionary participatory breeding populations because plants were removed from the participatory plots the first year. However, what we see from the results in our analysis of variance, the p-value of the breeding method is 0.27. They are not different. This could be because farmer negative selection acted on low yielding plants, or the act of plant removal stressed the neighboring plants and caused them to mature early. And the reason why I mentioned the benefit of early maturity in quinoa yield is because the trial had to be terminated and combined before complete plant dry down of the whole trial. This was the last surviving trial of 2015, and heavy rains were predicted. A major issue with quinoa is that there is little to no seed dormancy, and the trial would likely have sprouted if rained on. And so we see these low yields of populations 105 and 106, and then the parent lines, cherry vanilla and QQ065. These are all later maturing populations and parent lines in this region. Taking a look at the yields from 2016, the ANOVA produced a significant breeding method effect. That's fantastic, with the evolutionary participatory breeding yielding greater than evolutionary breeding. Populations 104 and 107 yielded greater than parent variety black. It's great to see that. It's important, though, to note that evolutionary breeding trials in other crops, they tend to have more than 6 generations of natural selection that has occurred to improve the agronomic fitness and outperform the parent lines. And in some cases, the heterogeneous populations were not suitable to become heterogeneous cultivars because the yield was not high enough and they were not stable. In such cases, it's suggested that populations should be continued for use as breeding populations from which individual lines could be derived from. And so that's what we have with our hedgerow trials. And so our hedgerow trials, we made selections using an index, a selection index. And the goal is to compare the selected lines. And we had to determine which traits were of interest. To do this, we created the survey that farmers completed before they conducted selection in the evolutionary participatory breeding plots. And we had the farmers rate traits from 1 to 5, with 1 being unimportant and 5 being necessary. We then added up these ratings as and used this as numeric weights for the selection index. So on the left of the table are the traits provided on the sheet in the survey, or they were also brainstormed by participants. And there's a column for all the added values that became the weight of the trait. The most right column are the traits that were eventually analyzed in the field and lab that could be matched with the traits in the initial rating sheet. And so remember, we're really looking to use this to determine top ranking selections. It applies weights to these important traits, and it's farmer driven because it's a survey that farmers filled out. So the results of the hedgerow trials, it's pretty exciting to see how the selection indexes is put to use. We have a table here showing results from the trial in Chimicum, Washington. At the top are the weights given to the traits directly below them, running from left to right, sorted by highest selection index first, so value 47. We have days to seed set, days to harvest, 1,000 seed weight, seed size in millimeters squared, days to flowering, and 3 different color attributes. So L stands for lightness, with 0 being black, 100 being white. A stands for positive value being more magenta and a negative value being more green, and B with a positive value being more yellow and a negative value being more blue. Next, we see in bold that are the trait values for cherry vanilla, which is a variety commonly grown in the region, and below that are the 2 other check varieties in this augmented design. And so now we want to know, what are the numbers and what are these stars? So in order to determine if a head-row trial was significantly different from the variety cherry vanilla for a particular trait, after running an ANOVA and determining if entries were significantly different, we then ran Dunnett contrasts to see what was significant. And we did this for all head-row trials. It was very exciting to see how we could then apply the selection index here. So SIS stands for selection index score. The selection for essentially when a trait of an entry was significantly from cherry vanilla, the selection index weight was applied. What we ended up with are 9 unique breeding lines suited to 3 distinct locations using this farmer-driven selection index. And numerous of those selections were from population 107 that outperformed cherry vanilla. So this is a success. This is pretty cool. So in conclusion, although this study captures only 2 years of evaluations of heterogeneous populations and only 1 year towards the development of pure lines, we see that the performance of population 107 yielding higher than one of its parents, and we've got EPB resulting in greater yields than evolutionary breeding. The most exciting is that we have 9 unique breeding lines suited to 3 distinct locations. Like, this is really showing that this method works. What's really great is there's so much more information about our methodology and our results. So you can learn more about this project. It's chapter 10 in this fabulous publication that came out in 2019 called Farmers in Plant Breeding, Current Approaches and Perspectives. There's so much more information that I couldn't share. And that's it. I'd like to thank everyone in my lab, everyone that was on my committee as my masters, and of course, all of the farms that were involved in this project. And that's it.
Thank you so much, Julianne, for such a great presentation. And we're going to open it up for questions now.
All right, I see a question in the Q&A from Tim. So he's asking.
Yes.
Oh, sorry.
I know, I froze for a moment there, so I'm just glad everything's working. Yeah, thank you so much for that presentation, and we'll go ahead and get started with questions. So if there's other questions, please post them here. Oh. So we have a question from Tim. Julianne, do you make the initial crosses manually? Do you use any molecular markers to exclude possible self-fertilization?
All right, the translation is in the answered column in the Q&A, and I will jump into answering that. So I did not make the initial crosses. It was a lab member named Adam Peterson who did the initial crossing event. He did do it manually and with the aid of a microscope, and he has a fantastic publication on his methods. I believe that they had used visual markers, so crossing to plants that have red leaves, so you can see if there was any self-fertilization.
Great, thank you. And we have another question here from Michael McElroy.
Muy
How
bien.
Go ahead.
Tenemos otra pregunta aquí de Michael.
How much seed can you get from the individual crosses?
¿Qué tanta semilla puede usted obtener de los cruces individuales?
How much seed can you get by the F2 or F3 generation per cross?
¿Qué tanta semilla puede obtener usted de los cruces individuales también?
Great. From the individual crosses, I do not have the numbers of quantification for how much came off of each event. By the F2 and F3, once they move out of the greenhouse and into the fields, it was really dependent on our success in the field of that year for the yield that would be coming out of those generations. Once you get to the F3, and especially in this evolutionary breeding model, we are seeing that the most successful plants will start, I guess, dominating in that population, and our yields have been increasing.
Muy bien, pues para los tipos particulares no vi mucha diferencia, pero en el caso de los F1 y los F2, depende en realidad de qué tanto éxito tengamos en el cultivo. Entonces, si tenemos una buena producción, vamos a tener buenas semillas, pero en realidad eso depende de qué tanto éxito tengamos en el cultivo.
We have time for one more question, it looks like it. We have a question from Dylan.
Tenemos tiempo para una pregunta más y esta pregunta viene de Dylan.
How did you evaluate taste? What was the correlation between flowering time and yield?
¿Cómo es que ustedes evalúan el sabor? ¿Cuál fue la correlación entre el florecimiento y la cosecha?
Evaluating taste has been one of our biggest obstacles in the breeding program.
El evaluar el sabor ha sido uno de los obstáculos más grandes en nuestro programa de mejoramiento de semillas.
We seem to have higher saponin varieties coming out, or the populations do have quite a bit of saponin.
Parece que hemos tenido bastante saponificación en las variedades que han salido, o al menos en la población de variedades que hemos tenido, tienen alta saponificación.
So I would absolutely love to collaborate with other researchers that might be doing lab or very small scale seed cleaning.
Así que me gustaría colaborar con otros investigadores que estén haciendo trabajo de laboratorio sobre la limpieza de la semilla.
Specifically the abrasions, that we can get some nice samples to then cook and do taste testing.
Ajá, especialmente en los procesos de abrasión, donde podemos tener muy buenos ejemplos para poder hacer un mejor testeo del sabor.
Thank you so much, Julianne. We still have a couple questions that we're going to move to Slack, where she'll be able to answer those for you there.
Muchísimas gracias a Julianne. Tenemos un par de preguntas que vamos a mover al canal de Slack, así que ustedes pueden encontrar las respuestas a estas preguntas en el canal de Slack.
And now we'll begin our next presentation with Blake Richard and Jared Seistro.
Y vamos a continuar con nuestra siguiente presentación de Blake y who was the other one? Can you remind me? Jared, perdón.
Yeah.
All right, well, my name is Blake Richard, and this is one of the fields of Wild Rose Farm, and we're in Humboldt County, and this in particular is in Blue Lake, which is about 5 miles inland from the coast, the ocean. And
I'm Jared Zeistro. I work with Organic Seed Alliance and have been working with Blake on some quinoa breeding and selection for the past several years here. I also live here in Humboldt County, but I work with farmers all around the country on seed saving, plant breeding, variety trials, and seed system building.
All right, yes, I've started growing quinoa in a small way, at least 15 years ago, just experimentally. And then, it seemed to work good, so we started growing a bit more. And we originally got our seeds from White Mountain Farm in Colorado, one of the original quinoa growers, obviously. And they had some really neat varieties. And then we kind of, we were just growing for the local market around here. And we kind of accidentally almost selected some of the weirder ones from their strains. They're white and they're black, and we ended up with a rainbow mix that we were growing and happy with. As far as a white variety, we started using Frank Morton's, which are so great, but we also had the idea that it'd be neat to try to kind of develop our own white variety with a big seed. Really had the interest of trying to get a big seed. And along about that time met Jared, and we started trying to select from the rainbow population big white seed.
Yeah, so it was at Eco Farm, at the Ecological Farming Conference. Blake approached me and mentioned his quinoa growing and the interest in trying to do his own breeding and seed saving. And going out to Blake's field, I saw a few things that really made it seem like there was a really good chance of having a successful partnership. When I'm thinking about participatory plant breeding and working with farmers, one of the things that I've first kind of heard as a recommendation to think about is when farmers are approaching you with a project, it's usually a really good sign, because that's something that they're carrying passion for and have a clear need. And Blake obviously had that, but also Blake had his own population that he'd been selecting and saving seed from and adapting to a somewhat unique environment. And I guess I can let maybe you speak to that, Blake, but compared to especially 6 years ago or so, how other people were growing quinoa in terms of spacing, but also in terms of kind of like how this is being produced, in the dry farm conditions, it seemed, like it was different than the way that other, for example, Frank's quinoa was being grown and selected.
Well, yeah, we originally started growing with pretty wide spacing in our quinoa, rows quite far apart, and actual plants, 6, 12, 18 inches apart. And we realized what a headache that was as far as weed control right away. Because, if you have a foot of row and you only have 2 quinoa plants and you have 2 weeds, those weeds are going to significantly affect the quinoas. And so we started getting higher and higher with our density in row, up to 10 seeds an inch kind of thing. And that also, we saw other benefits with that, where not only was it more competitive with weeds, but it also made for more uniform maturity and earlier maturity and easier combining. And so, that became part of the selection process, that plants, we needed to grow them in that way to see what was.
Yeah, so about 6 years ago now, Blake had already been saving his own seed from this diverse population that we already saw obviously had both phenotypic and genetic diversity. It was a population where he had been doing active selection, through natural selection, where it was having the plants were having kind of fairly intense intra-species competition amongst themselves to be able to compete. When 8 seeds per inch were being planted, not all of those were going to be growing and making seed, or if they were, some were going to be making a lot more seed than others. And so there was already natural selection for plants that could thrive and make seed in that competitive environment. Plus, you had already been doing some degree of selecting for larger seeds, just out, by selecting large seeds from your gravity table, right?
Correct, yeah.
So there'd already been this, what we call, recurrent mass selection in Blake's conditions out of this diverse population for several years. So there's this really good, strong starting population to work with. So about 6 years ago now, when Blake got started on, and I got started on a little more of this goal of saying, okay, it's great to have this diverse population, but it would also be nice to be able to have, as Blake had mentioned, like a large white variety and maybe some other colors, and be able to have them, you know, be separated out a little more while still retaining, you know, and maybe even enhancing some of these, you know, goals of vigor and, you know, adaptation to these dry farm conditions and these high, you know, high planting density conditions. So what we did was we went out to the field, and there was a really fairly nice field that Blake had grown that I guess actually was slightly less dense than usual. And one of the advantages of that was it was just a little easier for us to really identify individual plants, which can be sometimes the challenge when it's planted so densely to really see which plants look better than others. But this was slightly less dense, so that we were able to pick out and flagged around 50 or so plants that first year that seemed to be really promising, that also represent kind of a spectrum of colors, plant statures, because we, you know, we didn't want to lock ourselves into a particular, you know, phenotype right away. Save seed from each of those plants individually in separate little bags, so that we could basically use those as the start of trials and breeding the next year. So then now, 5 years ago, the following season, we had our first trial. And this was down in Lolita, and planted it in kind of a more traditional trial setup, where we had 4 rows to each plot, 3 replications. Actually, no, we had 2 replications of each variety, and then included a bunch of kind of control varieties in there from other, from some of the other seed companies like Frank Morton Wild Garden Seed, and looked at those, you know, taking each of those individual, what were individual plants, turning them into plots that next year, and looking at those plots as a whole and seeing which ones we liked. And I'd say some things that we learned that first year was that, I mean, it was nice to be able to see everything together. It was hard to get into those plots at that planting density to get stuff. It was hard to keep everything separated. And what we started to learn later on was it was also hard because the quinoa was crossing between those plots pretty actively, is what seemed like we were starting to see evidence of subsequent years. So sort of from that year on, there's always been this push and pull of balancing out, wanting to have lots of plots with lots of varieties, or lots of these families basically to look at, while still having enough space between the plots to try and minimize crossing, but recognizing that this is still, you know, valuable ag land, and not to waste all that space. And so all of those balancing factors have been some of the challenges in participatory plant breeding that we've been working through. But essentially, from 5 years ago through today, the process has been fairly similar, where we have identified the best, you know, the best plots, the best families, selected individual really prime looking plants out of those families from those plots, saving the seed individually from those plants, and then carrying that forward to the next year to become new plants, to become new plots in new, what we call family rows. And from there, always looking for, you know, in the case of the white ones, large seeds, earlier maturity, vigorous looking plants that seem to be yielding well. So yeah, each year saving seed from the best plants in each plot, and then taking those, in the seeds from those best plants in each plot, and having those become the new family plots, the new family rows the following year, usually looking for large, white-seeded, vigorous plants in the white-seeded variety. Also looking at, you know, some sort of red, or what we've been calling these sort of garnet, dark red looking varieties. And those have been kind of 2 of our main targets for variety types. But while we've been doing that, just because we don't ever want to go too far down a particular path and realize that there's some flaw, like, oh, this is now really ligase susceptible, or this seems to be too late maturing, we've been trying to bring in new material by selecting out of Blake's, what we call sort of general population, this vigorous population that he's been saving, as, a mass selection all these years. So each year we usually bring in a few of those into our plots as well. And so that's kind of what you're seeing around us now is, 6 years into this, more or less, you'll still see there's some off types in these plots. There's still some diversity, and I've at times I've maybe been frustrated that it's like, oh, we're making slow progress because of these off types. But at the same time, I think the advantage of that is we know we're still keeping some genetic diversity in all these varieties. And if we look more across the field, we see that over the course of years, we're kind of narrowing in more and more closer to our goals of these large seeded, more uniform, vigorous varieties, I feel like. And this year in particular seems like things are looking really well. But, you can kind of see in this field an example of the balancing act here of trying to, have a lot of plants, have, a lot of plants in each plot, have relatively large number of plots while still keeping some isolation between the plots and not wasting all the space. So, this, interplanting here of squash with quinoa allows us to be able to have a trial with, about, in this case, this year was a little lighter. Some years we've had 300 plots. This year, I think we have around 70 or so between here and down below. But have that many, on a working farm with large plots without wasting too much space. But in past years we've tried other things and they've worked to more or less success. I know, Blake, you wanted to talk to that a little bit and maybe make some recommendations for other, farmers and researchers that might be wanting to do participatory breeding.
Well, yeah, we've just figuring out where to put this stuff is what he's talking about. And we've definitely made a lot of mistakes with that. One year we put them all on the edge of the field, thinking, okay, on the very, very edge it'll be, kind of out of the way. But then the blackberries grew so much it kind of became a pain. Blackberries grew right through it all. And then another year we had big high sunflowers. We condensed the trials very in a small area, and we had sunflowers between them as a pollen break was the idea. But the sunflowers grew 12 feet tall and leaned inward, and you literally had to crawl through them to look at the quinoa. So yeah, just goofy stuff like that. Yeah, and it has been a little bit frustrating at times with the crossing that we're seeing, and we were a little confused, on just how easily quinoa crosses, and it seems like there's mixed opinions out there about that. But the ones we're growing definitely seem to cross pretty readily over, a bigger distance, a smaller distance than what was traditionally believed they did. And sometimes we wonder too, why even do this when Frank Morton has these great varieties and Lundberg's now developed a couple great varieties, and of course there's some other great varieties out there, but there's enough promise always to keep us going for sure with the idea of a bigger white and with the garnet we're working on. I like it. To me, these seed heads are going to end up looking real big, and I like the fact that the plants they look, other than the couple off types, the purple ones, they look very uniform as far as the seeds, the structure and everything. What do you think, Jared?
Yeah, I mean, I feel like, at this time of year, at least for me, first off, I defer to Blake on these things, and that's been one of the great things about this partnership is, Blake you've spent so much time, around quinoa for so many years that you have a really good feel for, what you like and what you don't like and why it might not work and why it might work. I guess for me, I feel like often this time of year, it's more a matter of looking at seeing problems, like seeing like, oh, this thing looks like it's really succumbing to disease or pests, or, it's got a lot, just way too many off types, and, seeing things that might be harder to see once it's fully mature. And then, feel like later in the season is a little more, there's a little more proof in the pudding once it's either harvest time and we see if something's going to mature in time, and then once it, all the way at cleaning, once we're actually being able to see the seeds themselves are, one of the things that we care the most about. So, this early in the season, we don't know if are these going to end up being, nice large white seeds, or are they going to end up being small, are they going to be kind of brownish? It's hard to tell this early in the season. So a lot of the time, like right now, it's more looking at what I don't like than knowing, that this is a perfect plant right now.
Yeah, I agree. The verdict's still completely out on all this.
Yeah, I think one of the examples we had was, we were really excited about a particular line that we'd found pretty early on.
Mud house, we called it
right,
mud house.
Yeah, mud house, because they had kind of this, it seemed to have it sometimes, and we weren't sure if it was maybe later on thinking that this might be, some insect damage that was causing the shape, but the kind of panicle was, ended up having kind of this slightly less of a pointed shape and a little more deformed actually, but seemed to yield really well, make large white seeds. So these, plants that seem to make, nice dense seed heads, nice large white seeds. We really liked it, and we, kind of blew it up for more of a, maybe like what was about an acre or something like that planting. And only then, once it was an acre planting put in amongst the rest of Blake's production, was it really obvious that it was like 10 days later or something like that compared to the general population.
At least. Yeah.
And so it was still all wet when it was time to combine and the rains were coming, and, it was like it was hard to see that when, it was in this smaller trial form. So that lesson really always sticks in my mind, where it's like one of the reasons we kind of maintain diversity here as we sort of push some of the populations towards seed increases, out of these trial fields into more seed increases to see how they look at larger scale. Like one, we want to keep doing that, making sure, how do they really look at scale once we think something is promising. These rows alone don't tell us everything. And two, knowing that we want to keep generating new diversity and not, put all of our eggs in any one basket or even any one family's basket, because it's really easy. Like I've kept track of okay, this was saved from, family number 3 5 years ago, and so was that, for example, and even though they're 2 different plots now because they've been saved from different families in subsequent years, they sort of all originated from the same plant 5 years ago, for example. Like I've tried to make sure that we've kept diversity in these trials the whole time and not, go too far down the road of narrowing that, because again, so once we get up to scale, sometimes you see things you don't see in the trials.
Yeah, we could just be on the wrong page altogether and just staying on the wrong page.
Yeah. So I think those are some of the lessons we've learned. It's been a really, a joy to work with Blake for these years, and
yeah, just so lucky to have Jared. I mean, what a resource, just his knowledge and just OSA and
Start that sentence again.
What an absolute, amazing thing to have Jared. What a resource, what a nice person, and OSA in general, just such a blessing to be able to have their help in doing this.
Yeah, I feel the same way about Blake. I mean, without, again, having participatory plant breeding is all about this relationship and all about the different things that different people can bring to the project, and being able to recognize that and leverage that, that Blake has so much expertise in this crop, so much expertise, in terms of, how to manage it in the field, to not only to grow good trials, to make sure that the trials are appropriately reflecting real agronomic conditions, and then maybe on the flip side, it's it's pretty easy for me to hold on to a spreadsheet and keep a bunch of seed, on a shelf over the winter and just keep stuff organized, because I do that for lots of other projects, and it, can be something you don't have to worry about as much. So I feel like it's been a good partnership.
Yeah, we just hope hope it ends up being real productive.
Yeah, right. In a couple of years, we'll see if we've got
Yeah.
some real winners that end up, available to farmers all around the country.
All right, yeah, so the way we plant these trials is just with a John Deere 71 planter on a toolbar, but we've taken out the seed metering stuff, the box on top, and we just have a big funnel. And Jared walks along and dribbles the seed in.
Yeah, I just have, if there's 50 or 100 or however many little packets of seed that have been saved from last year, it's just in a box, duct taped onto the toolbar, and I can just grab each packet and kind of, manually meter it in, dribble it in through the funnel down into the drill. And in the past, we tried using push seeders and things like that. And the nice thing about this is it's basically matches Blake's, it's the same thing Blake uses to plant his quinoa fields with. So kind of matching the system to some extent.
Plus, our soil here is a very heavy clay loam, and we have a duff layer on top that's pretty thick usually. So we have to plant the seed pretty deep. We often go 1.5 inches deep to plant to find moisture. It's all dry farm. We don't irrigate at all. We try to plant, as soon as we can in the springtime. We get a lot of rain here, of course, and so it's often a long time before we can get in and work the field. So we often don't get to plant until June. And so the planting date is always as soon as we can in the spring, which sometimes ends up being pretty late. Well, yeah, harvest I think is probably about 90 to 100 days after planting is about what it is. But of course that can be real variable depending on the weather. And also we've seemed to notice that the later you plant, the quicker it matures, but you don't get as good a yield as when you plant earlier. It takes a little longer to mature when you plant earlier as far as the amount of days. There's a couple of different ways we harvest. I mean, the trials, of course, we just harvest by hand and cut them and dry them and shake them into a bag. Our main fields, of course, we harvest with a combine, and sometimes we swath them first and get them with a pickup header, or sometimes we direct cut. The direct cutting is good when everything's dried down really nice, but if there's some green weeds in there, it makes sense to swath it so you can dry down the weeds also. Or if the crop isn't maturing very quickly in the fall, and but the seed is ready, the seed is mature, then swathing is good. Because you can cut it down and make sure you get it out before the next rain.
So a lot of times, like Blake said, for these trials, we'll go through and hand harvest. So maybe out of each plot, if the plot doesn't look good at all, we might not take anything from it. If it looks good, we might be taking 5 or 10 plants out of that family. And then those are going, sometimes more, but those will go basically into a bag that's labeled and then get dried more just in my office, and then I'll do the cleaning by hand, and kind of while I'm cleaning is when I'm taking notes on, like, okay, how well, did it seem like this plant, yielded, how large was the seed, how, what color was it, and make notes on which ones, kind of looked, I'll be looking at the notes that we took during the season for anything that we liked or didn't like about it, and then I'll be making additional notes as I'm cleaning the seeds. And based on that is usually when, we'll carry forward most of the stuff that we end up harvesting, because we'll be making decisions often as we're harvesting, but then stuff that's, as I'm cleaning, just obviously, oh, these are way smaller seeds than we thought, for example, or it turns out, they really look like there's a lot of seed in here, but once I was rubbing it out, there's just nothing here, then they'll get tossed and, won't be replanted. So there's sort of a final, negative selection that happens then, where some stuff gets pitched.
And it'd be really nice to do nutritional evaluations. That'd be wonderful. But yeah, nowhere near that. And as far as the taste test, all the quinoa from that we've ever grown always tastes great. And so we haven't been doing that. But maybe that'd probably be a smart idea too. But we have not been doing that.
Yeah, that'd be fun. I'd be interested to see the best methods for how to do taste testing. I'd be interested.
Wonderful. Thank you so much, Jared and Blake. We're going to open it for questions now.
Excelente. Muchísimas gracias a Gerald and Blake. Vamos entonces a ir a la sesión de preguntas.
Our first question is from Rural Action Sustainable Agriculture.
Nuestra primera pregunta viene de Rural Action Sustainable Agriculture.
A previous presenter was analyzing larger seeded quinoa.
Un presentador antes estuvo analizando un tipo de quinoa más grande, de semilla de quinoa más grande.
And said the protein content suffered.
Y dijo que el contenido proteínico sufría.
Have you thought about that in breeding for size?
¿Han pensado ustedes esto en términos de mejoramiento de semilla?
This is Jared, and I have not thought about considering the protein content in relative to size yet.
Este es Jared y tengo que decirle que yo no he pensado todavía en el contenido proteínico relacionado con el tamaño de la semilla.
I think that would be an important consideration and certainly something to monitor once, as we're, finishing out some of these varieties.
Yo creo que esa puede ser una consideración muy importante y algo que debemos considerar y estudiar tan pronto como estemos terminando con estas variedades.
Thank you. Our next question is from Gerardo.
Muchísimas gracias. Nuestra siguiente pregunta viene de parte de Gerardo.
And this question is for Blake.
Y esta pregunta es para Blake.
What is the vegetative period from seeding to harvest?
¿Cuál es el periodo vegetativo desde la siembra hasta la cosecha?
Blake, you might be on mute.
Jared, you can answer it as well if you know.
Jared, ¿lo puedes contestar si tú sabes? Parece que Blake tiene problemas con su micrófono.
Can you guys hear me now? Can you hear me now?
Yes.
Yes.
Okay, good. Yeah, it's about 100, 120 days, depending on variety, of course. There's a lot of range in the different varieties. And it also depends a little bit on what time of year you plant. And then, of course, it depends on the weather and how quick it grows. And one more thing about the nutritional content, that would be super interesting to do. And of course, there's a lot more, parameters other than protein content too to consider. I mean, maybe the protein content suffers, but maybe some of the other things are better. Who knows? I don't know if protein is the end-all objective. And so nutritional analysis absolutely would be really important to do down the line.
Muy bien. Pues le tengo que decir que el periodo vegetativo va entre los 100 o los 120 días. Eso depende de varias cosas. Por ejemplo, depende del tiempo en que siembra, depende del tipo de semilla, depende del terreno, depende de la variedad. Y también tengo que decir que con relación al contenido proteínico, eso es algo muy, muy importante que tendríamos que considerar, por supuesto. Blake, can you remind me the last part?
Well, the last part was just about the nutritional content, how protein content might not be the end-all objective always, and then maybe, there might be some other nutritional parameters which are better with any given variety. But nutritional, basically, I agree. Nutritional analysis is really important in the end.
Y la última parte es que el contenido nutricional y el análisis del contenido nutricional es muy importante, aunque debemos tener en cuenta que no solo es el valor proteínico, hay muchos otros factores y nutrientes, además de la proteína, que se deben tener en cuenta y se deben considerar. Pero por supuesto que es algo muy importante para considerar.
Excellent job, Sonia and Shirley, our translators. I just want to remind you all that as we are answering questions, to just give them a little bit of time to translate in between your thoughts. So We have another question here. Do some varieties seem to outcross more readily than others?
Tenemos otra pregunta aquí. ¿Algunas variedades son más fáciles de cruzar que otras?
This is Jared. So if any of the other experts in this panel have more specific information, feel free to jump in. My understanding is the short answer is yes. There's a range typically between 0.5% and 20% outcrossing has been found.
Si alguno de los expertos tiene respuesta para esta pregunta, los invito a que la compartan también, pero mi respuesta es que sí, que entre el 10 y el 20% de algunas variedades son más fáciles de cruzar.
Great, thank you. We have another question here from Rural Action Sustainable Agriculture.
Muy bien, pues tenemos otra pregunta aquí de Rural Action Sustainable Agriculture.
They are interested to know if you have anyone who has been successful growing for seed in the Midwest or South.
Ellos quieren saber si usted tiene conocimiento de. Can you repeat the question, Lauren? I can find it in the chat.
Yeah, sure. They're interested to know if you have anyone who has been successful growing quinoa seed in the Midwest or South.
Oh, ya. Si ustedes tienen conocimiento de alguien que haya tenido éxito cultivando la quinoa en el centro y el este del país.
And I'm assuming it's the United States.
En Estados Unidos.
This is Jared. And again, if anyone else, obviously there are a lot of experts here that have experience, so feel free to jump in. I have not, I've talked to many people who have attempted it unsuccessfully in the South. I have seen some quinoa being grown in at least the upper Midwest, so Wisconsin, Minnesota.
Otros de los expertos que tenemos aquí pueden también hablar y ayudarme a contestar estas preguntas. Yo en realidad no tengo conocimiento de alguien que haya tenido éxito cultivando la quinoa en el sur, pero sí algunos en el medio oeste.
And we have one more question here from Olivia.
Y tenemos aquí una pregunta más de Olivia.
And she asked, which distance would you recommend to eliminate crossing? More than 1.5 meters?
¿Cuál es la distancia que ustedes recomendarían para eliminar el cruce? ¿Más de 1.5 metros?
So, this has also been something we've been trying to work out, and it does seem, again, it depends, I think, on the variety or populations that you're working with, since some are more readily outcrossing, some seem to, really, essentially self-pollinate.
Bueno, esta pregunta depende en realidad de la variedad que usted esté sembrando o con la que usted esté trabajando. Hay algunas variedades que se polinizan a sí mismas y otras que son más fáciles o difíciles de que se crucen entre ellas. En realidad esto depende de con cuál variedad usted esté trabajando.
Based just on our personal anecdotal experience, it appears like we've seen crossing in our populations at distances of 1.5 meters. So, at least for, based on that, if your population is like ours, I would recommend greater distances than that to reduce outcrossing.
Muy bien, pues basados en nuestras experiencias en el cultivo, yo le puedo decir que nosotros hemos tenido cruces con distancias a 1,5 metros. Así que si usted en realidad quiere disminuir el cruce, tendría que plantar a distancias un poco más grandes, porque en lo que nosotros hemos visto, inclusive a 1,5 metros, se pueden cruzar las semillas.
Thank you so much, Jared and Blake. We'll move the rest of the questions that we have unanswered into Slack for you to be able to address them throughout the rest of today so that we can make sure all of our participants can see those answers. And now we're going to move on to our last presentation of this session with Junda.
Muy bien, muchísimas gracias a Gerald y Blake. Les queremos agradecer por su presentación y a ustedes les queremos decir que las preguntas que no pudimos responder las van a encontrar en Slack. Ahí pueden encontrar las respuestas a esas preguntas y pueden hacer más preguntas si es que tienen. Y por ahora vamos a continuar con nuestra siguiente presentación y le vamos a dar la bienvenida a Junda Jiang.
And nursery manager and breeder at Lambert Family Farm. It's a pleasure to present our research result. My title of this presentation is an efficient participatory quinoa breeding program in Lambert Family Farm. And I have main points today: why participatory quinoa breeding in Lambert Family Farm first, and second, how we do participatory quinoa breeding and what we have accomplished through this program. And it is mainly due to the vision from our leadership team, and with all the nutrition value of quinoa, this ancient grain finds its home in Lambert Farm. Our sales and marketing team found the niche market and consumers' interest, and our R&D are always innovative for new product development. And our agriculture department see potential for sustainable organic farming for this crop, and also see the growth interest. Technically, participatory plant breeding is an integral part of our operations for organic rice production and its continuous improvement. On-farm variety trial is a routine procedure before we release or use any new variety in our company. Growth and growth service manager actively participate in variety trial, including entry and location selection and crop management and even data collections. And the production team, QA and mill and sales market team and R&D team participate sometime lead milling, processing, cooking, and sensory nutritional quality evaluation. Quinoa is no exception to our operations. How we do participatory quinoa breeding and growth actively lead and participated in on-farm variety trial and make recommendations of variety allocation for commercial production. In 2013, Lambert Family Farm started the first on-farm variety trial in Litchfield. In 2014, nursery conducted the first replicate variety trial in Litchfield. In 2015, nursery conducted variety by planting date trials in Litchfield with Washington State University support. And in the same year, growth service manager Adam Peterson lead variety trial in northwest states. Excuse me. In the same year, Blake Richard participated in a Lambert Family Farm and Washington State University joint quinoa variety trial in Humboldt County. And this slide shows you how we set up a variety trial in Lolita, Humboldt. We do participatory quinoa breeding in Lambert Family Farm, and we have selected the best variety for organic white quinoa production in Humboldt and the Pacific Northwest. And we have variety from Frank Morton and we selected the best varieties, Chili Vanilla and Redhead. And then Blake Richard grows a rainbow-colored quinoa population for organic track quinoa production. So we don't have a pure line, pure black and pure red. So our sales and marketing team and R&D set up the breeding goals, and we need a pure black and a pure red quinoa variety for niche market. So in 2015 summer, we evaluated over 200 quinoa germplasm for desirable green quality and green color. And in the winter, and a quality assurance team evaluated 40 best black and red selections, and we did a greenhouse purity test. And in 2016 summer, we evaluated 170 selected progeny in 320 plots in Lolita, Humboldt, in Northern California. And we harvested 130 lines, 250 plots for yield and green quality data collection. Before we do that, before we harvest, we selected 500 heads for advancement and purification. Then in the 17 winter greenhouse, we evaluated 60 lines for purity test and selected the best 7 in Chile. And then we select the top 7 lines for bleed seed production in Blue Lake in 17 summer. And in 18, with the cooking test and the year data analysis, we select the top 2 lines, 1 black, 1 red, for foundation seed production. And in 2019 summer, we started the commercial production of red and quinoa variety in Humboldt. Let's show you in 16 winter greenhouse, we did the greenhouse purity evaluation in Chico State, University Farm Greenhouse. And we select the pure lines for advancement. And this slide summarizes 3 years breeding nursery in the Lolita in Humboldt, and 2 years in Lolita, 1 year in Blue Lake, and a school and nursery size about 2 to 3 acres. And you saw the lines we tested 170 16, 285, 235 18, and the plot number listed there. And we use different size plot and 20 feet by 20 feet long, 60 feet wide beds, and for 16, and then 15 by 5 square feet, and 10 by 5 square feet in 18. And this to show you the comparison also of how the percentage we harvested from nursery, and we applied the selection pressure highly in 17, and then in 18 we have 50%, 53% harvest, and the average yield also show difference. Okay, and so we select a lot of individual head for advancement, and this show you in 16 nursery, and left is a rainbow and the segregation, and the light is a pure line at the booming, pure black. At harvest time, you see the clear difference between the rainbow and pure black line. And before we bulk harvest, and we select the best head, and the larger head and the desirable brain color in both location. And in the winter, we did a purity test. We are very fortunate. We're happy to have Frank motivate us in our greenhouse in 17. And behind Frank is and John Schuver and Oriana as a technician showing the experiment. And this shows you how we harvest in the field. We cut by hand and split by hand. And so what we have accomplished, we developed two pure lines. One is called 18B67, a single selection from 16 plots. And then 17 we have a pure plots, and we harvest for bleed seed production. And then 17 we harvest for bulk harvested, and for 18 bleed seed production. And this time we grow 50 acres last year. And then we did a cooking test, and this show you our selection is superior to the market control in terms of the green appearance, and both dry and cooked, and aroma, texture, flavor, and also bitterness. And this is a 1 to 10 scale. And this shows you how we select our red quinoa from the one plot 16P205, we harvest only central row to improve the purity. In the greenhouse, we identify pure line for advancement. And this line has been tested in for all sensory score, and we found this line is superior also in terms of appearance and aroma, texture, flavor, bitterness, and overall score compared to 3 market control. And so this line is also a single header selection and from winter greenhouse. And then this line show excellent yield potential and green quality flavor texture from 3-year variety trial. And then we grow 2 strip in 17 for breeder seed production, and then 1.2 acre in 18 for foundation seed production. In the last year, we grew 30 acre for pilot organic production, and we achieved high yield, and this line is already now in market. So from 3 years with 7 cycle, and we successfully developed 2 pure line, 1 black, 1 red, and for organic quinoa production. And that is definitely the team efforts with marketing, sales team, the farmer, growth service managers, and quality controls station. And this is a testament to an efficient company-wide and growth participatory breeding program. And I have to thank my whole team and Aliana, Julega, field technician, and JP, research specialist, Steve Power, the assistant, and many student interns for their support. And also want to thank my supervisor, Jessica Lumber, and who led nursery from 13 to 19, and then Bryce Lumber, and VP of Agriculture, now he's leading nursery, and also the growth service manager, Eric Lumber, Adam Pearson, especially Adam for quinoa, and John Shuven, and Bill Carlson, and for their support and participation. And I thank Tim Schultz, VP of R&D, for his vision and support, and also for sales marketing team for their continued support. And it shows all the team members working in different areas from field to lab. And then I have to thank Dr. Kevin Murphy and his team from Washington State University Quinoa Breeding Program for their support, and moral support and technical support. And also to Blake Richard, Northern California quinoa growers, for his support in planting, management, and harvesting. Without his support, we cannot achieve this goal. And Frank Morton for providing wild garden, providing wild quinoa seeds. And also David Brenner for providing us new gene product from Iowa State University Plant Production Station. And also I want to thank Dr. Save Eric Jacobson for his advice, and I emailed him for advice many times for his generous support, for sharing his advice and experience. Certainly, I want to thank the quinoa community, and I feel fortunate to be a member of the community since 2013, the first Quinoa International Research Symposiums. With that, memorize John Shuven, our quinoa growth manager, and John actively participated in quinoa breeding program in planting, selection, and harvesting from 2016 to 2018. Thank you so much for your attention.
Junda, thank you so much for your presentation.
Junda, muchísimas gracias por su presentación.
We are going to take some questions now from our participants.
Okay.
Vamos ahora a responder algunas preguntas de nuestros participantes.
And just a reminder to provide breaks between your response for translation.
Y solo le queremos recordar que por favor hable un poco despacio o espaciado para que nuestros traductores puedan hacer un buen trabajo.
So Dwayne Johnson has a question for you.
Dwayne Johnson tiene una pregunta para usted.
First, he wanted to just let you know that your yields are amazing.
Lo primero que quiere hacerles saber es que sus producciones son impresionantes.
And his question is regarding the black quinoa.
Y su pregunta tiene que ver con la quinoa negra.
He wants to know if the pericarp has been removed.
Él quiere saber si el pericarpio ha sido removido.
Good question. You mean the sample we're using for cooking test?
¿Usted se está refiriendo al testeo para cocinar o a la cantidad que sacamos para probar, para cocinar?
I believe so.
Okay. Well, we using equipment we call Yamamoto dehuller. I'm sorry, not dehuller, miller. And we just mill 2 times, sometime more time. Remove the pericarp. Yes, pericarp. We did. Sometimes there's some trace, left, but we remove the pericarp. Yes.
Quiero decirle que sí, que con el método que nosotros usamos, sí removemos el pericarpio.
Great. Our next question is, oh, it just disappeared. Let me see here. We have a question from Jose. It's in Spanish.
Tenemos una pregunta de José, es en español.
Can you see it, Sonia?
Yes.
Okay.
Su pregunta es, ¿cuáles fueron los criterios de selección participativa a nivel de campo para las dos variedades consideradas? So, what was the criteria or the participative criteria selection in your fields for the 2 varieties that you presented about?
Oh, good question. We selected for certainly the yield and the quality. First, the quality, the brain color, seed size, and also the plant type, the maturity.
Muy bien, pues muy buena pregunta. Los criterios de selección fueron buena producción, maduración. Can you repeat the other ones, Dr. Jung?
Yeah, we selected for first the plant type. We look at a good plant type, elect and big heads and no lodging and overall the plant type and no severe disease infection.
Muy bien, entonces dice que la parte más importante es el tipo de planta y que seleccionar que no tuviera enfermedades.
Thank you so much for the questions. Do we have other questions for Junda? Looks like.
Muchísimas gracias por todas sus preguntas. ¿Tenemos más preguntas para Junda?
If not, you can always post questions on Slack as well for all of our speakers.
Si no, recuerden que siempre pueden poner las preguntas en nuestro canal de Slack para todos los presentadores.
I'd like to again thank all of our speakers for our session on participatory breeding.
Le quiero una vez más dar los agradecimientos a todos nuestros presentadores en el tema de mejoramiento participativo de la quinoa.
Thank you.
And I believe now we have a short break until our next session begins at 1:15, I'm sorry.
Muy bien, pues ahora vamos a tener un pequeño descanso mientras nuestra siguiente sesión continúa a la 1:15. Y aquí pueden ver lo que tendremos para nuestra siguiente sección.
Daniel Botero, would you like to test your mic for the next session?
Okay, let's see. Do you hear me?
I can hear you great, thank you.
Perfect. Hi, thank you. So, and Maria Bruno is connecting. She just sent a WhatsApp message.
Okay, we will promote her from an attendee to a panelist.
Okay. So Maria goes first. I'm going second.
Let me double check how we have it set up. Yeah, you'll be first.
Okay. Yeah. Okay. So it's the opposite of the title, but no problem.
I'm here as well. I don't know if you can see me.
We see you, Maria. How are you?
Good. I guess I'm looking at the. Okay, I'm fine. How are you, Danielle?
Fine. Had to stop for a while.
I know. I haven't been able to. I've just been busy with our classes started this week, so it's been hard for me to be there
I
live.
remember you were not sure you would be able to be at this time, so it's good.
Yeah, I knew I would be able to come, but I knew that I was not going to be able to maybe see all the other presentations.
Yeah.
But I responded to one of your questions this morning. Did you see that?
Yeah,
on Slack?
I saw people who work in Quinoa sometimes used to repeat the same idea time and again, and we don't test that. And the only reference I found for someone who prohibit quinoa cultivation is in that book of the one from the archaeologist in the University of Santiago, this lady, I can't remember her now.
Who's the cultural anthropologist?
Yes, that well, the one
Carmen.
The lady who studied the archaeological seeds from the central Chile.
Maria Teresa.
Maria Teresa. You know her book.
Yes.
That book. In that book, there is a reference about one publication saying that quinoa was forbidden at the time of the colony. That's all I found. I was never able to access that publication. But when you read about quinoa in the past, what you have is reference which are positive about quinoa. And not negative.
Also I was thinking of Hunziker wrote that big thing about how they confused quinoa and amaranth.
Yeah.
And how the category of food kind of got fuzzy. So
yes, but okay. If you speak about amaranth, I agree that amaranths were used in a bloody ritual by the Aztecs. So that's another issue. But I don't think there is a clear connection. So your interpretation is, for me, more probably.
Yeah, I think it was more gradual than that. I think it was a food that the Spanish thought was weird, and so they didn't really take it on.
Yeah.
And then I think, as you say, as soon as market forces kind of come into the equation, farmers are making decisions about what not only can they feed their family, but then how they can enter into that.
Yeah, but Eileen Caparelli, she was the PhD director of Laura Lopez.
Yes.
Argentina. And they published an article. I share the link in Slack. Around 15 years ago, and they were doing research in a place in the province of Catamarca, and they found wheat and barley
really early
associated to ritual places. And at that time, the Spanish were not there yet, because there were these Calchaquí wars again, so the Spanish couldn't go to that place. So that indicates that people got the seeds.
Yeah.
And if they use the seeds, means that they were not under the pressure of the Spanish to grow it. And the general attitude of the people in the Andes is open.
Yeah.
They always
And you see that
try new things.
everywhere. I mean, European trade goods make it across the Americas well before
Yeah.
people do.
Yeah.
So yeah, that's
But I don't. Yeah, of course, I don't have the time to do that. It's not my profession, but it could be a good subject.
Yeah.
To do some research.
There have been a couple dissertations, one in Bolivia and another in Peru, that look at that early colonial period, looking at the plant remains and how those crops are coming in and other ones are going out. And I think I don't think you can say like across the board, the Spanish prohibited it and that's why it went away. I think every location has a slightly different story of how those things kind of came and went.
There were, of course, many things which change with time. For example, something that I remember for reading the books at the time of the conquest, there was no racism. What I mean with that, that the Spanish were very clear who they had in front, and they were very respectful of their enemies. You know, racism came later.
Right,
but
when they brought slaves.
Yes, so once you are the dominant side, then racism comes with domination, but not at the start. So in many ways, there was a period of, you know, testing each other, understanding each other, seeing what each other had to offer, and coming closer in time. When I work with the people in the northwest of Argentina, they don't make a difference between quinoa, barley, faba bean, etc. They consider all these plants their plants. You know, these plants have been there for 300-400 years.
Oh yeah, absolutely.
So you have to be an archaeologist to make the difference.
Yeah, well, I mean,
this is native, this is not native.
Right. Well, just look at plato paceño, right? It's corn. I don't know if you've had this when you were in Bolivia. Faba beans, cheese, and potatoes. So I mean, yeah, definitely.
Yes, it's a combination. Yeah.
I'm gonna grab my headphones in case, I don't know, sometimes the volume isn't very strong. So I'll be right back. I'm gonna grab my headphones.
Okay, see you. Okay. Telling some
Welcome,
of my
Daniel.
colleagues
Welcome,
that I'm going to be first.
Hi, Laura.
So I'll just do a short introduction since we have recorded introductions.
Right.
Okay.
And just a reminder, as you're responding to questions, to leave time for the translator to translate after each like sentence or short.
Okay,
response.
Laura, if you prefer, I can do both. I can answer in English and Spanish as you want.
That's great.
Yeah,
Okay.
I could too, unless, you know, you already have the translator set up, but either whatever's easier.
Yo soy la traductora, pero por favor siéntanse libres de hacerlos porque a veces hay mucha terminología de laboratorio con la que yo no estoy familiarizada. Entonces esa se hace un poco difícil.
Listo.
Perfecto, perfecto. Bien, vamos viendo y nos ayudamos mutuamente.
Muy bien.
Perfecto.
Estoy de acuerdo y apoyo a Sonia.
So Laura, Ivan told me I go first and Maria goes second.
Yeah, that is fine.
Okay. Because if you put it in time, she has to go first. Because it's in the past. No problem.
We can switch it. It just depends if
no, it's fine.
If you have a preference for answering questions.
Yeah. You know, I asked so many questions, so maybe now is the time for revenge.
So I do have another, I know we talked about this in the preview, but should we be monitoring the questions or should we wait till the end and answer them kind of all at once?
We will read the questions as they're being upvoted to you. So they will ask questions kind of throughout your presentation. Hopefully people will be posting questions.
Okay.
And then we will ask those questions of you. And then any that we don't get to, we'll post to Slack.
Got it.
And then
Okay.
you can answer them there.
Okay.
Yeah. Maria, what I have seen is sometimes during presentation, you will receive some questions.
Mm-hmm.
So maybe you can start looking at those questions at that time,
Preparing
and
them.
then wait for Laura or whoever to ask the questions, and then so we have to respect the order they ask the questions, but we
Right.
can look at the question before.
Great.
So this is fine, but it's not the same as being together in
No.
person. There's no replacement for that. You were in the first meeting, Maria, or not? I'm not
No,
sure.
I don't think so. The only one I've been to is the one in Ecuador. That was
You went to Ecuador, yeah.
2013. Mm-hmm.
Yeah, because you know we started in the meeting and then we finished the night in a bar or a restaurant until very late. You cannot do that this time. But anyway, it's going very, very well.
Yeah,
So congratulations to the organizers.
it is.
Yeah, it's been really wonderful to see the last 3 days and how everything's come together. So
Yeah, it's not. In fact, we were organizing a meeting in China. Not so many people, just a small number of speakers, but it was going to be in June. Then we decided to postpone it to November, and now it's highly probably we are going online.
yeah,
Because in this year it will be impossible, because you know now there is no coronavirus in China, but they don't want us to go there now. So there are restrictions for foreigners to go there. So I don't know for how long this is going to be this way. Maria, do you have to go to Bolivia or Peru?
Well, I hope so. We have an NSF grant. We were supposed to go this summer, but obviously couldn't go. And
Yeah.
who knows what will happen next summer. Fingers are crossed that we can go, but
Yeah.
we'll see.
Well, you know, here in Argentina,
it's good.
Illegal to go out of your house.
Oh, really?
You have a strong reason to do that. If you go to the supermarket, you have to go to the one which is near your house. Only part of the population is allowed to walk. So if you are part of that population, then you can have a permission. And when the police stop you, you show that permission. But if you don't have that permission, they can take your car.
Oh my gosh.
And that's complicated because you don't know when you're going to have your car. And the faculty, all classes went online. And that was for the first semester. We are starting the second semester now. And the second semester is going to be online again. And some people say that maybe we are not going to be able to go back until next June. The point is I had to cancel. Yeah, I had to cancel some of my experiments this year, but I cannot keep going this way. I have experience in the summer, so by January, I need to start. Okay. So we are starting?
Yeah, we're going to go ahead and get started.
Okay, so do you want us to mute or just you?
Yeah, you can mute until, yep, that sounds great.
Okay.
Evan's going to go ahead.
Okay, so video and mute. See you.
Okay, everyone, welcome to the session this afternoon.
Hola, muy buenas tardes a todos. Bienvenidos a esta sesión de la tarde.
As participants, you do not have the ability to share video or sound, so please use the Q&A box for questions during the presentation.
Como participantes, recuerden que ustedes no tienen la habilidad de compartir sonido o video, así que recuerden que solo pueden participar escribiendo sus preguntas en la caja de preguntas y respuestas.
And use the thumbs up button to vote for questions that you would like to be answered. You can vote for your own question as well.
Y use el botoncito de me gusta para votar por preguntas que hayan hecho. Ustedes incluso pueden votar por su propia pregunta. Así les haremos estas preguntas a nuestros presentadores.
And after each presentation, there will be a Q&A session.
Después de cada presentación, nosotros vamos a tener una sesión de preguntas y respuestas.
And after that session, any unanswered questions will be moved to the Slack channels for further discussion and conversation.
Y después de esa sesión, cualquier pregunta que no hayamos podido responder, la vamos a mover a nuestro canal de Slack. Allá van a encontrar respuestas y también más discusión sobre estas preguntas.
And now I will turn things over to the facilitator for this session, Laura Lewis.
Y ahora le doy el mando de esta sesión a Laura Lewis, que es nuestra facilitadora para el día de hoy.
Thank you, Evan, and welcome everyone.
Muchas gracias, Evan, y bienvenidos a todos.
This panel is from the domestication and diversification of quinoa to present-day water and nitrogen use efficiency.
Este panel se trata de la domesticación y de diversificación de la quinoa hasta el presente, la eficiencia del uso de agua y nitrógeno.
Dr. Maria Bruno will be speaking about the archaeobotany and evolution of quinoa throughout the Andean region in South America.
La doctora María Bruno hablará sobre la arqueobotánica y la evolución de la quinoa en la región andina de América del Sur.
She will discuss current knowledge around progenitors as well as suspected progenitors to domesticated quinoa located throughout the Andean range.
Ella va a hablar de las evidencias y teorías alrededor de sus progenitores durante el proceso de domesticación y discutirá varias metodologías utilizadas hasta la fecha para comprender la evidencia arqueológica relacionada con el cultivo, uso y diseminación de la quinoa en toda la región.
She will be discussing various methodologies used to date to understand archaeological evidence related to the cultivation, use, and dissemination of quinoa throughout the region.
Y como lo dije, ella va a discutir varias metodologías utilizadas para comprender la evidencia arqueológica relacionada con el cultivo, uso y diseminación de la quinoa.
Daniel Bertero will be speaking about nitrogen use and water efficiency in quinoa.
Daniel Bertero les hablará sobre el uso del nitrógeno y la eficiencia del agua en la quinoa.
He utilizes both ancient varieties and domesticated populations to improve the crop for various types of sustainable production systems located throughout South America.
Él utiliza variedades tanto antiguas como domésticas con el fin de mejorar el cultivo para los varios tipos de sistemas de producción sostenibles ubicados en América del Sur.
We will be hearing from Daniel Bertero first with questions and answers, and then we'll be hearing from Maria Bruno.
Muy bien, pues entonces primero vamos a empezar con Daniel Bertero y preguntas y respuestas y luego veremos la presentación de María Bruno.
Welcome to our second speaker, Daniel Bertero, for our session from the domestication and diversification of quinoa to present-day water and nitrogen use efficiency at the International Quinoa Research Symposium. Dr. Bertero studied biology at the University of Cordoba in Argentina with a PhD thesis in agricultural sciences at the University of Buenos Aires, where he worked on quinoa's photoperiod and temperature responses. For almost 30 years, he has been working as faculty of agronomy at the University of Buenos Aires and the National Research Council on several aspects of quinoa ecophysiology, seed physiology, germplasm collection and characterization, and genotype by-environment interaction patterns. His research group has been able to identify sources of pre-harvest sprouting resistance, which are being introduced into breeding lines. In addition to showing that quinoa grown in highly productive environments is limited by its capacity to produce more seeds, more than by the capture of resources. thereby opening the door to the manipulation of reproductive partitioning to increase yield potential. Daniel has contributed to safeguarding Argentina's local genetic resources together with the knowledge needed for its exploitation and management. Please join me in welcoming Dr. Bertoro for this great session.
Thank you, Laura. This is the second time I participate in the Quinoa meeting organized by Washington State University. It's a great pleasure to be there again. At that time, it was an intense period of a lot of discussion and exchange between the people who was there. Before I start my presentation, what I want to say is that I'm not going to show results of our research. This is part of a review, so I use data from other people's work, and a significant part of the data I use for today's presentation comes from undergraduate student theses in different countries of the Andes. And why do I want to say this? Because these were very good research studies, and they have not been published, so I'm really happy to be able to share their results. So going back to my presentation, as you see in the title, the question I'm trying to answer is whether quinoa is efficient in the use of water and nitrogen. We work with quinoa, and we know from research that quinoa is regarded as an efficient crop, most in the use of water. We'll speak about nitrogen later. And when you speak to farmers, they will say that quinoa is a species which use less water than other crops. But the research we know mostly have been done at the plant physiology level, and today we want to show what we see when we go to the crop level of understanding. And how did I start with this? Last year, I had the opportunity to travel to China, to Shanghai in particular, to have a sabbatical, stay there, to do some experiments at the plant stress center. The plant stress centers belong to the Chinese Academy of Science, and I had to wait for the plants to grow, and then I had time to write a book chapter. I was asked to write the book chapter about quinoa physiology, and this book chapter is going to be part of this book. This book is going to be the third edition. It's called Crop Physiology. It's pretty known for the people in the area, but for the first time, if you look to the right bottom side, you see a photo of quinoa growing in South Bolivia. So, and that means that quinoa can be in the book speaking about major crops. So I think most of us will be happy to see that is happening, and that is also in parallel to what is going on with quinoa in the world today. To start with my part, we are perhaps pretty familiar with this kind of slides in which we have different levels of organization. They will have different disciplines, and each discipline is based on a particular level or range of level of organization. So when we speak in terms of plant physiology, usually we are speaking about plants growing pods and looking at processes at the plant level, or maybe at the organ level like leaf, or maybe at most basic levels like tissues, or even smaller levels. But when we speak about crop physiology, we are looking at the crop. That means we are trying to study and understand what happens with the population of pretty homogeneous plants of the same species growing in a rather homogeneous environment, which is plot. So that's the difference between what is plant physiology and what is crop physiology. So crop physiology is some way in between what is physiology on one side and what is ecology on the other side. Before doing this or writing this chapter, I went to Google Scholar and looked for information about quinoa in the last almost 20 years. And then these are the results just for writing quinoa. as you see, there has been a steady increase in the number of publications with time. And if you look at the arrow, you see that marks the year when the International Year of Quinoa was celebrated. I think that in some way that contribute to the increase in the number of publications. So that is for quinoa as a whole. But then when you go to physiology, particularly to the different stresses to which quinoa is faced, you see how those papers are distributed by subject. So most of the papers related to quinoa physiology and stress comes when you write drought or water. So there are more than 150 papers published on quinoa from that period. Second comes salinity, third comes nitrogen, and then you have temperature and heat. And to the right, you have the combination of these different stresses. So as usual, there is less information about this combination of stresses. But if we think that quinoa was a species which was pretty much unknown 30 or 40 years ago for science, today we have a lot of information. So quinoa became a model plant for water stress responses, became a model plant for salinity responses. And then going to the crop level, this is a photo from an experiment which is being going on right now in China. I will divide my talk into two parts. First, we'll speak about water, and in the second part, we'll speak about nitrogen, and I will finish just with some comments about how we can make some progress in dealing with these two stresses. This photo is from Buenos Aires, and this is what water means for us. So in Argentina, water regularly means a lot of water, at least in the part of the country where I live. Usually for quinoa, we think in terms of water for water stress. For us, the problem with quinoa is not the lack of water, but the excess of water. But this is not what I'm going to speak about right now. How do we approach water from an ecophysiological perspective? This equation was posed by a scientist from Australia almost 40 years ago. His name is John Pasura, and he used this equation. So he divides the different components related to water and affecting yield in terms of, first, the amount of water which is used by the crop, which is crop evapotranspiration. Second comes the proportion of that water use, which is used as transpiration, so what the crop effectively used. Then comes the transpiration efficiency, which is the ratio between the biomass accumulation by the crop and the water transpiration of the crop. And finally, we have harvest index. So this is the approach I'm going to use for this presentation when speaking about water. French and Schultz were two scientists, Australian scientists, and took the task of analyzing how yield is affected by the availability of water. These papers were published in the 80s, so some time ago, and the novelty of what they did is that they define what's called yield boundary to the response of yield to water. And it shows what the role or the place of the relevance of water in limited yield in places where we used to say that they are places where water limits yield. And this is kind of circular. Well, but the point of this is this. If you look at a figure like this one, this is the relationship between yield and crop evapotranspiration for quinoa. The relationship between any 2 points is also called water productivity, which is the relationship between yield and water use. There is a maximum slope of 13.3 kilos of grain per millimeter of water use. So that is our maximum response. That is the limit we get today for quinoa. All other points are points that for several reasons are not directly related to the limit posed by water. That can be disease problem, can be frost problem, can be mismanagement of water, of sowing dates, etc. And another message coming from this relationship is that the idea that quinoa use little water does not seem to apply to all conditions. If you look at the right, you see some points with very, very high water use. And in the box to the right, what is shown is values for this maximum slope, what's called a water limited yield, for crops like sunflower, maize, and wheat. So for sunflower, it's around 8 kilos per hectare. For maize, it's around 42. And for wheat, here they are reserved for wheat in Australia, for wheat in the United States. So as you can see, save for sunflower, the values for quinoa are pretty low. When we analyze how these points are distributed in geography, what you can see is that the maximum values are those coming from temperate environments. In particular, these come from experiments which were run in Denmark in the spring, summer, and in Morocco from the autumn to the spring. So those are the maximum values we get. And if you look at the right, you can see the result from Iran. This comes from one experiment which was run in the summer there under very high temperature and irrigation. And this crop used more than 1,000 millimeters in crop evapotranspiration, and of that, around 1,000 was used by quinoa in transpiration. That's a lot of water. So with that water, quinoa was able to produce on average around 3 tons. But 3 tons, we were pretty much expensive in terms of the use of water. And finally, most of the points below the 1 ton value come from Andean countries. So it shows that something is going on in the Andes, making the quinoa crop not able to use the water which is available to the crop as well as it could be used. So the question is, as the question says here, who are we going to blame? So according to the equation, it can be related to the partitioning of water to transpiration, can be related to transpiration efficiency, and could be related to harvest index. Today, I will just speak about transpiration efficiency. And this slide shows the relationship between transpiration efficiency and crop evapotranspiration. Again, I will remind that transpiration efficiency means how much biomass we produce per unit of water use. And why did I use crop evapotranspiration? What we should do is, like in the figure to the upper right side of the slide, those are results for wheat, which what we have is water use efficiency, which is the same to transpiration efficiency, against vapor pressure deficit, so the water demand of the atmosphere, because it's pretty well known that the higher the demand of the atmosphere, the lower the water use efficiency. In the case of wheat, it goes from around 80 kilos per millimeter to around a little bit more than 20 kilos. In the case of quinoa, we have that information just for a few crops, a few experiments, sorry, but we can trust that basically the relationship we see here is pretty much related also to variation in the water demand of the atmosphere. And it can be seen that the values of transpiration efficiency go from around 30 or 35 kilos to around 10 or maybe less kilos. So again, as in the yield against evapotranspiration relationship, which is shown previously, the best results are those coming from the north of Europe and Morocco, and the worst conditions are the ones we get to the right in Iran, but also in the Andes. When we compare these values from quinoa with the average values of other crops, what we see is this, that the transpiration efficiency here is shown for wheat, maize, rice, sunflower, and canola, also named colza. The values for quinoa on average are close to rice, sunflower, or canola, but they can be pretty low, and as I showed before, much lower than those from wheat under some conditions. So the point is, do we insist or do we focus on water use efficiency in quinoa? That's an open question, but I want to show these results. These results come from experiments done by Juan Gonzalez and his team in Tucumán. And to the left, what is shown is the relationship between photosynthesis and the time of the day for several quinoa cultivars, most of them coming from the highlands in Bolivia and Peru. And what is seen is that while some of these varieties are able to keep higher photosynthesis during the day, some of them are not able to do that, so photosynthesis go down around midday. And to the right, what is seen is the relationship between photosynthesis and leaf conductance, and these values are for midday. And what came out of this paper is that those varieties which were able to maintain photosynthesis through high leaf conductance, which also mean that they will have higher transpiration rates, were also the ones which had the higher leaves and the higher biomass. And what is particular of this experiment is it was not done under full irrigation. There was some limitations to water availability, and even so, the ones which were able to use more water were the ones which finally were more productive. And that brings us to one paper which was published several years ago by a person named Abraham Bloom, and the title of his paper is perhaps the effective use of water, and not water use efficiency is what matters in terms of productivity of crops under drought stress. And there are other services of transpiration. We are working on this right now, but basically, if a plant has a higher transpiration rate, then also have a higher cooling capacity, has lower temperatures, and that can also help to improve the productivity of plants under hot conditions. And that is part of research we are doing right now. So my second part of my research is related to nitrogen. The approach we use to nitrogen is based in these two equations. So yield can be understood in terms of the amount of nitrogen which is uptake, the nitrogen uptake of the crop, the nitrogen harvest index, which is the proportion of the nitrogen which goes to the seed, and finally the percent of nitrogen in the grain. If we use the same information, then we can estimate which is called nitrogen uptake efficiency, and also is called physiological efficiency in other papers, which is the relationship between yield and nitrogen uptake. And if we do that, this efficiency depends on two factors. On the one hand, the nitrogen harvest index, the higher the harvest index of nitrogen, the higher the efficiency, and on the other hand, the percent of nitrogen or protein in the seed. In this case, the higher the content of nitrogen, the lower the efficiency. Using the information from the Foliar publications, I was able to make this figure in which what is shown is the relationship between yield and nitrogen uptake in the black cycles. This is the first time we have this relationship for quinoa, so I'm really happy that there was information to do that. Some of the points are below the top value, so this is also a kind of boundary response. And on the right hand of the figure, what is shown is the nitrogen utilization efficiency, and this is shown by triangles. So for quinoa, I can go for around 60 kilos of yield per kilo of nitrogen uptake to less than 20. So when we move from less fertility to high fertility, there is a strong decrease in the efficiency of use of nitrogen by quinoa. If we go to the first component of this relationship, this shows the relationship between nitrogen utilization efficiency and nitrogen harvest index. There is a very strong relationship, and the arrow shows that when we go from lower productivity to higher productivity, the nitrogen harvest index decreased strongly, and also does the nitrogen utilization efficiency. If we go to the second component of this efficiency, then this is the relationship with grain nitrogen concentration. There is a tendency, the tendency is still significant, but it's pretty weak. And this other figure shows the relationship between nitrogen utilization efficiency and grain protein concentration under high productive environments for several crops. This is information from the literature. And then we have maize, we have wheat, we have sunflower, and we have soybean. And we have also quinoa. So there is a general negative relationship, which is what we expect as we increase the protein content of the seed. The minimum values then are for soybean, but soybean has around 80% of its nitrogen in the seed. The other crops have between 70 and 75%, but in high productive environments, the value for quinoa are around 50%. So quinoa under high nitrogen availability has a limit to translate the availability it gets from the soil into the nitrogen which goes to the seed. And we can call that the nitrogen harvest index gap. If we increase this gap, we can keep the protein concentration of quinoa, but increase the efficiency, and also we can increase yield. So where we go from here, so what's next? And you know, quinoa is grown today in many countries, so there are new environments, but we need new ideas. Or maybe we can borrow some old ideas for a new crop like quinoa. We have done something related to that. These slides shows the relationship between yield and growth per plant after flowering, coming from several experiments. The line shows the one-to-one relationship. And what is seen here is that the higher the growth, the lower the proportion of that growth which goes to yield. So when you analyze the limitation for yield, this is called a sink limitation. So there is a limit capacity to translate higher growth in higher yield. And one way to solve it is to do the same, which was done during the Green Revolution in other crops, which is to manipulate the gibberellic acid metabolism in a crop like quinoa. So we did an experiment. This experiment was published by a student named Gomez, her surname, and she applied paclobutazole. Paclobutazole is a gibberellic acid synthesis inhibitor, and see that at the beginning of reproductive growth. And this photo shows how the control plants and those treated with paclobutazole look at the end of flowering. So a lot of biomass went to the panicle, and that biomass came from the biomass of the shoot, as in other crops. So this also was related to a high increase in harvest index and final yield of this material. But it had another benefit, and the benefit was that also roots grew bigger under paclobutazole. So maybe we have a win-win situation. So we can have a higher yield on one side, but also we have a higher capacity for root exploration in a crop with quinoa if we work on manipulating the gibberellic acid synthesis or sensitivity of a crop like quinoa. So to finish my presentation, which are the main messages and questions coming from this? First, we could increase yield response to water and nitrogen if we increase yield potential. And also, if we manipulate gibberellic acid, perhaps there's also a benefit in terms of root growth. Second, that we need to solve the crop versus plant and leaf level propagation efficiency paradox. So I'm not saying that the results from physiological experiments are wrong, but there's something which is not seen in the same way when we go to the crop. This data come from different genotypes grow in different environments, so we need to pay more attention to genetic variability. Third, we need to work on filling this water and nitrogen limited yield gap. So perhaps not focus so much in the upper level, but what we can do in between. That's what many farmers and breeders have been doing in other crops, so this is not new. Four is very important that we increase nitrogen harvest index under high nitrogen. And finally, we need to do more research and focus of breeding on nitrogen. About nitrogen, we really know very little, mostly related to fertilization responses. So thanks for your attention. And now I'm ready to answer the questions which come out from this presentation.
Daniel, thank you so much for your presentation. We are going to go ahead and open this up for questions now.
Daniel, muchísimas gracias por su presentación. Vamos a empezar con la sesión de preguntas y respuestas.
Just looking here. Oh, our questions just went away. Just give me a quick moment. Let me see here. I have a question from Evan Crane.
Yeah, I can see it.
What are some traits breeders could use to indirectly select for nitrogen and water use efficiency?
¿Cuáles son algunas características que se pueden usar o que los mejoradores de semillas pueden usar para seleccionar indirectamente el nitrógeno y la eficiencia en el uso de la eficiencia y el nitrógeno? El agua, ay, perdón. ¿Cuáles son algunas características que los mejoradores de semillas pueden usar para indirectamente seleccionar el uso y la eficiencia del nitrógeno y el agua?
Hello, do you see me?
I can.
Yes.
Okay, I don't see myself. About that question, I understand I answered it partly in the second half of my talk. I will speak in English first and Spanish second. So my idea is that if we have kind of a green revolution for quinoa, so if we manipulate gibberellic acid metabolism, we will do as we know from other crops. So we'll increase the partitioning of photoassimilase to the seed, and we will reduce the height of the plant. And how is that related to nitrogen use efficiency? In other crops, we have some information about that for quinoa too. The higher the harvest index, the higher the nitrogen harvest index, and also the yield of the crop. Ahora voy a español. En relación a esta pregunta, yo creo que en parte la contesté en la segunda parte de mi charla. Yo creo que necesitamos una revolución verde de la quinoa en el sentido de manipular el metabolismo de ácido gibberélico, reducir la altura de planta, aumentar la partición a granos. Y eso, ¿cómo se conecta con la eficiencia de uso de nitrógeno? Eso ocurre a través del índice de cosecha. Si aumentamos la proporción de la biomasa que va al grano, eso de alguna manera tracciona la demanda de nitrógeno del grano, aumenta lo que se llama el índice de cosecha de nitrógeno, y eso aumenta el rendimiento sin modificar el nitrógeno absorbido por el cultivo, y eso hace que el cultivo sea más eficiente.
Excellent, thank you. Our next question comes from Ingrid von Behr. Daniel, the relationship of the protein in the grains and nitrogen. Have you studied the relationship with sulfur?
Hola Ingrid, no solamente, de hecho, nunca he trabajado mucho en cuestiones de nitrógeno. Esto es tenemos una estudiante que estudió esto, pero no sé si haya mucho más para decir con quinoa y azufre. I'm going to say this in English. Ingrid is asking if we also did some research on the use of sulfur and the impact on quinoa. We have not done that kind of research, and I'm not aware that kind of research has been done in quinoa. Of course, I know from experience with other crops like wheat, for example, sulfur is a limitation for wheat here, and if you combine sulfur with nitrogen, you get higher yields, but not for quinoa.
Our next question is from Raimondo, and he asks, what evaluation technique of efficiency of water use do you recommend?
In my experience, the simplest tool to evaluate water use efficiency is carbon isotope discrimination. But I have been reading for many years the papers by John Passure, etc. And in many cases, what makes a crop more efficient is not water use efficient. I'm sorry about the kind of confusion of what they say, but you have to be efficient in the use of water as a whole for the crop, and not specifically at the efficiency in the use of the water at the leaf level. But definitely, carbon isotope discrimination is a good tool to do that. En español, la pregunta es, ¿qué herramientas uno puede usar para estimar la eficiencia de uso del agua? Y entiendo que una de las mejores herramientas que tenemos es la discriminación isotópica de carbono, ya que hay una relación muy estrecha entre la discriminación de carbono por los estomas y la eficiencia de uso del agua, que a veces es una relación negativa la que tenemos. Igualmente, eso es una eficiencia a nivel de hoja, y lo que entiendo que debemos hacer es mirarlo a escala de cultivo. Muchas veces existe la noción de que un cultivo es más eficiente si es más eficiente a nivel de hoja, y tenemos que considerar otros aspectos, como por ejemplo la capacidad de cultivo de extraer más hoja o de reducir la evaporación, aumentar la transpiración del cultivo, y eso la discriminación isotópica de carbono no lo van a mostrar.
Thank you. We have time for one more question, at least. And here, this is a response from Ingrid with sort of another inquiry. In other cereal grains, the gluten content increases and gets better with the application of nitrogen plus sulfur.
Mm-hmm. This is not a question. This is just a comment.
Yes. Let's see here.
We have no gluten in quinoa, so
That's right.
I don't know what to say about that. La pregunta de Ingrid es acerca de la relación entre el azufre y el gluten. O sea, como dije antes, no sé sobre específicamente azufre. Quinoa no tiene gluten, o sea que si hubiera que buscar un impacto, habría que buscarlo por otro lado.
Great. And we have another question that just came in that's in Spanish by Javier. Do you want to read that, Sonia?
Yes. La pregunta dice, ¿ha estudiado la eficiencia en el uso de otros nutrientes además del nitrógeno, potasio, fósforo? The question says, have you studied the efficiency in the use of other nutrients such as nitrogen, potassium or phosphorus?
Comienzo en español. Personalmente nunca estudié ninguna de esas eficiencias. De hecho, parte del trabajo que hicimos fue una tesis de grado de una estudiante. Hay muy poca información sobre respuesta a nitrógeno, como comenté antes, y entiendo hay mucho menos con relación a otros nutrientes, y la información que hay es de respuesta a fertilización, no acerca de mecanismos fisiológicos. In English, the point is whether we studied the response to other nutrients besides nitrogen. I don't do research on this in particular. I have just the experience of one undergraduate student who studied the distribution and efficiency of the use of nitrogen in Buenos Aires here doing her thesis, but I'm not aware of much work on other nutrients, and the information available is just for yield responses to fertilization. There's nothing in my understanding about the mechanism involved.
Thank you so much. We're going to move on to our next speaker, and any of our unanswered questions will go into Slack for Daniel to be able to address.
Okay, thank you, Laura.
Muchas gracias a todos. Cualquier pregunta que no hayamos podido responder, la vamos a mover a nuestro canal de Slack, así que allá pueden encontrar las respuestas o hacer más preguntas para Daniel. Y vamos a continuar con nuestra siguiente presentación.
It's my pleasure to introduce our first speaker today, Maria Bruno. Maria is an associate professor of anthropology and archaeology at Dickinson College. Her research examines the long-term dynamics of indigenous agricultural systems in the Lake Titicaca Basin of the Andes, using ethnobotanical and archaeobotanical methodologies. A major component of this research includes the study of the origins of agriculture in the region, specifically the domestication and diversification of quinoa and Quinoa. This is accomplished through analysis of seed morphology in modern and ancient specimens using various microscopy techniques. Great, well, welcome and looking forward to hearing your presentation.
So thank you everyone for including me in this great international session on quinoa. I'm very excited to be able to share some of my research and work of some of my other colleagues about what we know from the archaeological record about the long history of quinoa in the Andes. So a little bit of an overview of my talk today. I'm going to start with just a short discussion of how we study the histories of crops through different methodologies. And then I'm going to focus a little bit on what archaeology is and the subfield of archaeology known as archaeobotany, in which we examine archaeological plant remains and has been the main way in which we've been able to investigate the history of quinoa. I'm going to talk a bit about what we know about quinoa domestication, and I'm going to focus in particular on what we know about its domestication in the Andean region, in the high Andes Mountains. And then I'm going to kind of zoom in a little bit more in the region that I work in, the Lake Titicaca Basin, which is a high lake that sits between the two ranges of the Andes Mountains between Peru and Bolivia, and talk a little bit about what we know about when quinoa was adopted there and the trajectory of its use in what we would be considering prehistory and even a little bit into more historic times. So there's, in the past, I don't know, 25, 30 years, we've made some really amazing strides in how we study the origins of agriculture and the domestication of crops and animals around the world. And many of the participants in this session this week are people who've kind of been pioneers in the study of using molecular genetics and the the relationships, the evolutionary relationships between living plant populations, such as the living ancestors of quinoa, the quinopod that was domesticated in North America, and then down into the Andes. And so these botanical and molecular approaches to the origins of any crop around the world, but particularly quinoa, have been really helpful for us in understanding what the progenitor populations was. So who was the ancestor of our domesticated quinoa, which botanists identified as being Quinopodium hircinum. And more recent genetic work has sort of verified that and then shown how that species was likely domesticated, possibly sort of independently in both the highland Andes region of the Andes, and then another species known as Quinoa del Mar on the coast of Chile. So this botanical and molecular evidence really is helpful for us for pinpointing sort of where these processes took place, what species were involved, and then can get us obviously thinking about the phenotypic changes that come with crop domestication, but also those underlying genetic ones. And my colleague, Dr. Botero, will be speaking of some of that work in terms of understanding other aspects of the plant's physiology. Archaeology has always played a really important role in understanding domestication, even before we had the technologies associated with molecular biology. Archaeologists, beginning in the late 1800s, early 1900s, began recovering plant remains from archaeological sites across the Andes. So it's great to be able to see these ancient plants look at what they look like in the past, maybe how they've changed over time and into the present. And perhaps more importantly, in terms of figuring out how this process occurred, is we can radiocarbon date large plant remains. And that gives us the most precise idea of when these things would have occurred. So while molecular science can kind of give us a little bit of clock in terms of like when genetic changes how long they may have taken to occur, those are often rooted in the dates that we get from archaeological sites and archaeological plant remains. So today I'm going to focus on what archaeologists have been able to uncover about the processes of quinoa domestication using these archaeological plant remains. And then the second part of my talk will really speak to how finding where these plants occurred and the kind of broader social and political context in which these plants were used and modified gives us a better insight into why people would have shifted to using domesticated plants and some of the other cultural and environmental factors that were going along in terms of people changing the ways that they were living in the world by depending on domesticated species. Okay, so this is a picture of our archaeological project on the shores of Lake Titicaca in Bolivia in the Southern Lake Basin. So archaeologists, again, I think any impression that you might have of them coming into this symposium is that we dig in the earth, and we certainly do that. And here we are working with our colleagues, our Bolivian colleagues, as well as our Aymara colleagues who are the residents, continue to live and cultivate these lands and then participate in the archaeological projects with us. So the subfield of archaeology that specializes in the plant remains that come out of these archaeological sites is known as archaeobotany. And we have a wide range of plant remains that we can examine. I'm going to be focusing today on what are known as macrobotanical remains. So you can see here in this picture, and I'll explain why in a minute, why we really just focus on those little black plants. Those are plants that have been carbonized, and because they're carbonized and kind of neutral, they can last in the ground for thousands and thousands of years without decomposing. So macrobotanical remains normally consist of wood, tissue of tubers, and then of course seeds, which we'll be focusing on. Plants also make other kinds of cells that will preserve archaeologically. My Argentinian colleague, Pilar Babot, has studied, these are some images of quinoa starch grains that she's identified on tools that they have recovered from sites in Argentina. So these starch grains or phytoliths can also be recovered from soils and on tools. Some of these cells may not be as susceptible to changes related to domestication, and they are also a little harder to date. And so really it's very helpful to have macrobotanical remains because we can see those changes and then date them when we find them. So just to give you a little sense of how we do this, as we excavate, every context that we separate out as we're excavating, we take this is at least where I work in the Lake Titicaca Basin, it'll vary from region to region, but we take about 10 liters of soil from every specific context, and then we put it through what's known as water flotation. So we have this system of water, buckets and water that's floating in, and we dump that soil into the water, and the light things float up to the top and come over the little water flow here. And a lot of those things are burned plant remains. We also capture some stuff in another screen that's below here, which often has like fish bones and maybe larger pieces of plants. So then we take that sample, which becomes very small from that big 10-liter bucket of dirt, and then we can look at it under a stereo light microscope. And again, like I mentioned, in this region of the world, only carbonized things preserve. On the coast of Peru, for example, or Chile, where it's extremely dry, you may get just desiccated plant remains. A colleague of mine, Laura, who works in southern Bolivia, they find in caves uncarbonized quinoas, which are really amazing. But what I work with mostly in much of these older sites are carbonized remains. So we sort out the different types of species that we find, and then we can look at them more specifically. All right, so I'm going to turn to quinoa now. I've included a song. So some colleagues of mine, Denise Arnold and Juan de Dios Yapita, have recorded some of the traditional songs that Aymara farmers sing as they prepare their crops. And one of the songs sung for quinoa is warank waranka, which means thousands of thousands. This plant, as everyone here I'm sure participating knows, this plant produces an abundant number of seeds on each of its panicles, and they get everywhere. And so I have this picture of a woman threshing her quinoa, and this is good for archaeologists, right? These seeds are kind of durable little entities. They go everywhere, whether or not you want them to or not. They are very likely to get into a fire, and because of that, they are very abundant in the archaeological records. So this is just a picture of all of the quinopods that we've picked out of one of those samples. And you can see the variation that is here in what's left in those seeds. So thankfully, quinoa is actually just a wonderful species to study its domestication because it is ubiquitous. So, and by that, I mean, it appears in almost all of the samples that we encounter, at least in the Titicaca Basin and in a lot of other places where it's produced. So it occurs across many different kinds of archaeological contexts. And then it's dense in the fact that we usually get many more than one little seed. We get hundreds of seeds in any given sample. And they're large enough in size that once we've studied that seed, we can send it off to a radiocarbon date, to an AMS date, and actually get the very seed dated. So we can know exactly, or within a range of radiocarbon range, how old that plant is. All right, so I'm going to turn now to how archaeobotanists can study the domestication of quinoa and the things that we look for in the morphology of these seeds that allow us to determine if the seeds that we're looking at are wild or if they've been domesticated. So again, this is probably no mystery to the people participating in this symposium, but quinoa presents itself with a lot of different variation, right? Even in wild plants, you'll get seeds that are black and a few that are white. But in the cultivated varieties that we know, they tend to be light colors such as white and yellow, and maybe red. But the wild quinoa in the Spanish-speaking world or in South America is known as quinoa negra because it's black. It has a black seed, whereas the cultivated one tends to be a lighter color. When we look at the morphology that creates the difference in these colors, there's a lot of actually other different things that we can look at. So one of the changes over a long period of time that we might see, and this is with several other crops as well, is that the seed tends to get larger. So humans, as they selected for certain species, they often want more food for what they're, right? So they're selecting for bigger grains. In actuality, there's not a huge difference, or at least statistically, between some of the domesticated species and the wild ones. So that doesn't seem to be the most important attribute that we can look for identifying a domesticated seed. But the other things related to the seed coat and some of the technical terms, the outer seed coat's known as the testa, the texture of that seed coat, and then the shape of the seed itself seems to vary with the thickness of that seed coat or the testa. So what we can do as archaeologists, and this is, I should point out, a method that was established first in North America as archaeobotanists there were figuring out that the North American indigenous peoples had actually domesticated a species of quinoa or Quinopodium as well. And they looked at comparisons between modern wild quinopods and domesticated ones. And the main change that we can observe with a scanning electron microscope and zoom in on it is the thickness of that outer seed coat or the testa. So this is a modern wild quinoa negra. You can see how thick that seed coat is versus this is also again a modern quinoa seed domesticated, and it's just paper thin, it's testa thickness. So as archaeologists, we can identify these seeds that might be a little more beaten up and maybe the whole testa is not preserved, but we can zoom in on it and measure the testa thickness. And study after study has shown that the wild species have a thicker testa than the domesticated species. So generally speaking, again, this will vary from variety to variety or species to species in different parts of the Americas. But in the Andes where I've worked, we've found that domesticated species tend to be less than 10 microns. And in the case of that Quinoa del Mar, it may have completely lost that outer testa. So that's really interesting to see. And then as you can see in this picture, even without the scanning electron microscope, you can begin to see these differences between the wild and domesticated seeds. So these little wild quinoa negra seeds really pop out. They still appear in agricultural contexts, because these would have been weeds that were growing alongside the domesticated species. And then the seeds here that look a little smoother, those are the ones that are likely the domesticated ones. And so several colleagues of mine have been working on this in different parts of the Andes. If you want to read more about that, I'll point you to an article that I published with Maria Teresa Planella and Laura Lopez, my colleagues in Chile and Argentina, where we summarized some of this work that's been done in different parts of the Andes. What I want to focus on now a little bit is what we know specifically for the domestication of the Andean species, because this is really where I would say we still need a lot more work on this question. It's certainly not completely answered, but the work that's been done so far in terms of identifying morphologically domesticated species, so these seeds that have thin seed coats and then have ideally they've been directly radiocarbonated, this is what we have. So you may have read studies of some quinoa seeds found in the north coast of Peru that date to a bit earlier, about 5000 BC, but those have not been subjected to the more detailed morphological analyses that I'm talking about here. There were a series of caves that were excavated in the Junin region of Peru in the early 1990s. Carol Nordstrom was a graduate student, was sort of one of the first people to apply this use of SEM, scanning electron microscopy, to look at the differences between wild and domesticated quinoas. And she identified domesticated varieties that dated to a level in the archaeological site that was about 3000 years before the current era. So the seeds themselves weren't domesticated, but they came from a layer that dated to that time. Subsequently, work by myself and Felissa Eisentraut in a site in northern Peru and sites in southern Bolivia, or around the Lake Titicaca Basin, we both identified morphologically domesticated quinoa seeds, and they were directly dated to 1500 BCE. Interestingly enough, the reason I bring up why it's important to actually date the seed is because the seeds were found in a layer that was dated with wood that were about 3000 BCE at Calcatani, Peru, but when they actually dated the seed, it was a bit younger. So these little seeds can move around in archaeological sites. So this is why we tend to put some stress on actually trying to date the seeds themselves or the object that the thing has come from. So based on that, there's been some interesting ideas about how quinoa and why quinoa was domesticated in the Andes, and work by my colleague Deborah Pearsall, who did the initial study of those seeds from the Junin region, as well as some other scholars have proposed that those who study the domestication of camelids see that those animals were actually probably taken under control and domestication before plants were. And as we know, so I have a little picture here, these camelids, llamas and alpacas eat the plants around them, and they also eat quinoa. So this is actually a seed in quinoa dung that's from an archaeological site. So there may have been this sort of co-evolutionary relationship where wild stands of the progenitor of the domesticated quinoa was not only attracted to the disturbances that humans were creating in the places that they were living, but that these animals were also kind of bringing those plants into the lives of the people who were harvesting them. So based on our radiocarbon dates, we can imagine that people were beginning to collect and manage wild quinopods as they were moving their plants around, or their herds of llamas around. And then around 3000 or 2000 BCE, they would have started to domesticate them. And then they become these fully domesticated plants that people transition to depending on, right? So we go from people gathering these wild species and using them as food to then planting them and kind of committing themselves to a really change of lifestyle where they become farmers, right? Where they're committed to these crops, they plant them, they care for them, they harvest them, they become less mobile and more wedded to the particular landscape. And so the more we've been thinking about this, and my colleagues and I working on the Taraco Peninsula have been looking at where we find these things, where we find domesticated quinoa show up, it often is with fully domesticated camelids and also even potatoes. So in the Near East, they've often talked about this sort of Neolithic package where wheat and barley and then eventually sheep and cows become domesticated and move around and spread to other parts of Eurasia. And maybe perhaps we have similar kind of agropastoral package in the Andes that includes camelids, quinoa, and potatoes. So I'm going to spend the last bit of my presentation here talking about what happens, particularly in the Lake Titicaca Basin, as we look at the use of quinoa through time, about up until the Inca conquest, when we've had quite a few archaeological projects in this region that have systematically collected plant remains and can track these things through time. So as I mentioned, the earliest dates that we have, both from the northern part of the lake basin and the southern part of the lake basin, are about 2,000 or 1,500 years before the current era. And that often includes the appearance of domesticated camelids and tubers. So we consider these people the first agriculturalists in this region. And it happens to coincide with the time prior to this. This lake basin was actually very, very cold and dry for several thousand years. But the climate seems to have ameliorated around this time. And that it made kind of the perfect place for people to settle down and become farmers. So the main time periods, I'm going to kind of, this is again just kind of a brief overview, but as you can see here, what we call the formative period is this really long period between about 1500 and 1500 BCE and 200 in the current era, where lots of things changes. But this is where we consider, if you're comparing this again to the old world, kind of our Neolithic, where people become farmers and become increasingly dependent on that. Their societies also, populations grow and become more complex. And this results eventually around 300 current era, the rise of the Tiwanaku state, which is the first large kind of political entity in this part of the Andes. So we've been able to track the use of different crops across this time. So our project, the Traco Archaeological Project, has mostly focused on the formative period. So when you look at these sample numbers, you'll see that our sample sizes for the formative periods are much larger from the Tiwanaku. But what you can see is, first of all, ubiquity is the percent in which we find these species in our samples. So it means that quinoa is present between 80 and 100% of our samples almost across every time period. So and parenchyma is storage tissues. So we can't say what kind of tuber this plant might be coming from, but you can also see that tuber tissues are quite prominent in all of our samples. We do have this little bit of a drop here in quinoa. I think that has to do with our sample size. I'm going in my next slide, I'll show that quinoa did stay very important food even during the Tiwanaku time. But what I want you to take away from here is quinoa increases important through time. Tubers seem to increase later in time, but they're kind of this important staple crop. These samples come from my colleagues who've worked at the site of Tiwanaku itself. And you can see up here, right, in all of the sites that they've studied, all of the samples they've studied, quinoa is present in over 90% of them. So it is literally everywhere. But what's interesting is you see this continued increase of parenchyma or potentially tuber cultivation. And then here comes maize, this crop that originally domesticated in Mexico, but then makes its way into the Andes quite early, and then eventually up into the highlands. So quinoa and tubers really are a staple food throughout the formative and the Tiwanaku period. Even in the Tiwanaku period, when maize, this kind of fancy crop, makes an appearance, probably to be drunk as chicha, these foods like quinoa are the kind of still the important foods feeding populations. So my last example is from this later time period called the Pacajes, or the late intermediate period. So the Tiwanaku state collapses and these communities develop into sort of more independent, autonomous chiefdoms or señorios, you may have heard of as that. So my colleague Brianna Langley worked at a site excavated by Liz Arcouche, and this is in the northern part near Puno. So people were living at the top of these outskirts, or these hilltops, during the late intermediate period. And what they found in excavating their house compounds were just literally millions of quinoa seeds. Brianna would show me like a giant Ziploc bag filled with quinoa seeds, ancient quinoa seeds. So what's really remarkable is this time period is kind of thought to have been maybe more a focus on just herding camelids, because you could kind of keep them up there. But what they did was terrace this entire hillside and cultivate quinoa and some tubers, absolutely zero maize, in this interesting time period. So with the conquest of the Incas and the Spanish, of course, this, any kind of conquest kind of impinges on people's kind of livelihoods to some extent, particularly with the Spanish. new crops were introduced. So in the Titicaca Basin region in particular, faba beans and barley become really important crops. The haciendas, so the sort of landowners from the city who owned the agricultural lands that the indigenous people still populated, tended to focus their crops on things that they could sell in the city. So potatoes and chuño, and again, maybe even maize and faba beans. But so when in the 2000s, when I've been working on this region, quinoa is definitely less prominent in these Titicaca Basin communities than it once was in the past. Other foods have replaced it. But as you can see from this woman who has her nice little stand of drying quinoa, people still produce, grow it, every other year for household consumption. And it's still a crop that people know about and enjoy, and often reminisce about how before the introduction of bread and things like that, it was the food that they ate every day, that their grandparents ate every single day. With that, I'd like to conclude by thanking the various institutions that have supported this work, and my colleagues along with the Traco Archaeological Project, and of course the communities that have participated and taught me so much about quinoa. And finally, I would like to dedicate this to two Bolivian scholars who, without their support and help, I would not even be speaking to you here today about this. One is who recently passed away. Juan de Dios Yapita was an Aymara linguist, and he was fundamental in translating a lot of the interviews that I conducted. And Eduardo Pareja was an archaeologist who helped me get the permits to do the projects that I'm presenting for you today. So along with the indigenous communities that have brought us this amazing crop, I want to recognize these two scholars. Thank you.
Maria, thank you so much for that presentation, and we have some questions for you. The first
María,
question,
muchísimas gracias por su presentación y tenemos algunas preguntas para usted.
the first question is from Evan Crane.
La primera pregunta viene de Evan Crane.
And he wanted to know if you found evidence of saponin removal in the work that you're doing as well for preparation.
Yeah, so I saw that. Oh, do you want to ask the question first, Sonia?
Él quería saber si usted había encontrado evidencia antigua, como de los indígenas, del removimiento de saponina.
Well, so interestingly, as I showed in the pictures here, most of the seeds that I get are carbonized. And if I'm not mistaken, most of the saponins are sort of in that pericarp, so one of the outer layers of the seed. So it has many layers of kind of seed coats. And I rarely find any seeds that still have any remnants of the pericarp on it. So that suggests that probably they're removing it. But also my colleague Laura Lopez has done some ethnoarchaeological work in the southern part of Bolivia, where she kind of documents the ways in which washing the grain and grinding it and boiling it and different kinds of food preparations serve to remove the saponins. And then she sort of looked at what the seeds look like after those processes have taken place, and has been able to kind of identify some of those things so she's worked with samples in southern Bolivia that are not carbonized, where you can maybe see some of the pre-processing that goes on of the seed before it's eaten. So I think there's more work to be done on that, but I think, I didn't mention this in the talk, but one of the processes we think about during domestication is detoxification. So that's particularly pertinent when we think about potatoes, but it would have also been the case for quinoa with the saponins on the outside. So it could be that later in time they were selecting for less bitter quinoas, but they certainly could have been soaking and grinding and cooking them in a way that would have reduced it as well. And I would refer everyone to Laura's work about that.
Do you feel prepared to repeat that in Spanish now?
In Spanish, yes. So, bueno, el problema que tengo yo en las muestras que tengo de Bolivia es que la mayoría de las semillas son carbonizadas. Entonces, muchas de las saponinas se encuentran en el pericarpio, una de las capas externas de la semilla. Entonces, en las muestras que tengo, que he estudiado yo de lago Titicaca, casi ninguna tiene el pericarpio. Casi ya ya se fue. Y eso puede ser por el hecho de haber procesado la semilla a través de remojar en agua o moler, hervir, que ha sacado un poco el pericarpio y los saponinas. Pero como las semillas son carbonizadas, es un poco difícil de distinguir. Sin embargo, tengo una colega argentina, Laura López, que ha estudiado los procesos de quitar las saponinas con los agricultores y cocineros de hoy día en el sur de Bolivia, y hacen estos procesos, ¿no? De remojar, moler. Hay diferentes preparaciones para diferentes comidas que tiene el efecto de quitar las saponinas. Y ella ha estudiado muestras no carbonizadas de quinoas antiguas en Lipes también. Y ella se encuentra evidencias de del procedimiento de sacar el pericarpio y las saponinas. Y un punto más es que el proceso de quitar estas cosas tóxicas, creo que no, los químicos tóxicos que se encuentran también en la papa silvestre, seleccionar para plantas con menos saponinas o menos tóxicos es una parte importante de la domesticación de la planta. Pueden seleccionar a especies con menos de estas cosas o pueden procesar la planta misma para quitarlas.
Thank you. Our next question is from Julianne Kellogg. How do researchers believe camelid livestock consumes here it is again, quinoa or quinoa browsing, or is it possible that they were fed the grain directly?
That's a really interesting question. I'll answer it in English and then I'll ask the question and then repeat it in Spanish. So the question of whether people fed directly their animals plants or like foddering like they do in the Near East is a really interesting one, and one that actually we've debated in the Andes for quite a while. We think, it's very likely, especially in the earliest things, that animals are just out there eating the plants. Obviously, you would want to keep your animals out of your growing fields because they can destroy them. But I think what we're finding probably really early on, animals are just eating the sort of wild, maybe partially managed stands of quinoa and wild quinoa, and through stopping at the campsite and pooping in their corrals would have encouraged that plant to grow. So once the animals were domesticated and the plants were domesticated, there would have likely been great effort to kind of keep those things separate. But what I've observed in my own ethnographic work in the Andes today is that even though farmers do their very best to harvest like everything that they can from their fields, potatoes and all kinds of plants get left, and then the animals are put into the mostly harvested fields to graze. And so they are grazing on the weeds, but also probably the quinoa plant that didn't get harvested or the potato plant that didn't get harvested. And I have with my, so we don't think that they're growing any of those crops to feed the animals. The only plant that maybe they were feeding to animals is possibly the totora reed that grows around the lake. But we mostly think the animals are just kind of getting access to the plant after the field has been harvested. Bueno, la pregunta era si hay evidencias para alimentar los animales domesticados, como las llamas, con quinoa, o si están comiendo las plantas en sí misma, no sin como una alimentación a propósito. Y la evidencia, bueno, pensamos que en las épocas iniciales de la domesticación es que los animales están comiendo las plantas silvestres y casi domesticadas de quinoa mientras que están pastoreando. Y es este proceso de comer estas plantas silvestres o semi-domesticadas que los animales están trayendo también estas poblaciones de plantas a los asentamientos de los humanos. Entonces, no pensamos que al principio la gente estaba alimentando los animales con plantas como quinoa. Tampoco hay mucha evidencia que estaban cultivando las plantas para alimentar los animales. Lo que yo he observado con los agricultores hoy en día en la península de Taraco es que sí, mantienen las plantas y los animales aparte, pero una vez que cosechan los campos, los animales ponen los animales en los campos cosechados para comer lo que queda. Y normalmente, aunque intentan recoger todo lo que han sembrado, se queda algunas plantas de quinoa, algunas plantas de papa y los animales comen eso. Entonces, no tenemos mucha evidencia de que estaban cultivando plantas específicamente para los animales, ¿no? Pero quizás estaban cosechando la totora, ¿no? Esta planta que crece en las laderas del lago Titicaca para dar a los animales. Pero pensamos que la mayoría de estas plantas domesticadas que entran a los estómagos de los animales llega a través de un accidente o si están comiendo después de la cosecha en sus campos.
Great. We have another question here from Mark Samuels. Can you comment on the timing of Chenopodium berlandieri occurrence in archaeological sites in North America compared to Mexico and South America?
Oh, good. Okay. Oh man, I hope my graduate advisor, Gail Fritz, isn't watching this right now because I have to remember all these dates. Yes. So in North America, the process of domestication seems to occur a little bit later. So it's what is known as the late Archaic period. And so that would be around like 3,000 years ago. So it's a little bit, I've got to look that up, but around, I believe the dates for Chenopodium berlandieri domestication is sort of between 3,000 and 1,500 BC. So not too different than what we're finding in the Andes, but the processes there are quite a bit different. In Mexico, the evidence that they have, so interestingly enough, the work, I would have to look that up because I don't remember it off the top of my head. But I think it is also a bit later, like around 2,000, 1,500, but I will have to look that up because I don't have that off the top of my head. La pregunta es los fechados de domesticación para los Chenopodium que se ocurren en Norteamérica, el Chenopodium berlandieri y también las Chenopodiums domesticadas en México. Y de lo que me acuerdo, y quizás puedo buscar rápidamente esta pregunta, tengo algunos datos aquí, pero de lo que me acuerdo es que la domesticación de los de Chenopodium berlandieri es entre 3,000 y 1,500 años antes de Cristo en Norteamérica, y que en México es un poco alrededor de 2,000, 1,500 también.
Great, thank you. We have another question here. Are you ready for a couple more?
Sure, if we have time. Yeah, great.
Yeah, great. A question here from, let me see. I think we have one that's already been translated. Just bear with me. From Mike Dutchinson. We have some SEM work in trying to identify seed coat thickness, but we are having some trouble interpreting the photos. It would be great for us to show them to you for your input. Perhaps, as you say, some of our varieties have a very thin or non-existent seed coat.
Yes. Yeah, so the question is that they, bueno, la pregunta es que hay unas fotos de Chenopodium arqueológico de otras partes. Do you remember where they said they're from? Did they mention?
No, I don't believe so, but we can connect you on Slack.
That's great, yeah. Pero es a veces difícil de encontrar interpretar la foto, porque como hay muchos niveles de capas de semilla, es difícil decir si está la testa o otro capa que hay ahí. So the question was, sí, mándame sus fotos, podemos verlas y ver si podemos distinguir qué capa tiene. La otra posibilidad es que se ha perdido completamente su testa, ¿no? Y como he mencionado, parece que eso ha pasado en Chile, en la costa de Chile, Pero también en Norteamérica hay, parece que en algunas de las variedades de Chenopodium berlandieri que esta capa también se ha desaparecido, ¿no? Entonces se han seleccionado para que no aparece para nada, no la testa. So I'll say that in English. One of the problems is that, well, there's a lot of different layers of the seed that could be measured. And so you want to make sure you're measuring the outer layer that seems to change with domestication. But it is the case where some Chenopodium domesticated seeds lose that outer layer completely. So one example does seem to be that Chilean domesticate, but also in Eastern North America, they've found that some of and they have uncarbonized, perfectly preserved seeds from caves that do not have a testa, so they've lost it completely. So that does make it a little bit more complicated, especially if you have a carbonized seed. It's hard to tell, did it lose the testa just getting banged around and processed, in preparation for cooking or after it got burnt, or was it that way when it grew? Entonces, depende de si las semillas que tienen están carbonizadas o no. Porque el proceso de carbonizar y comer puede causar que la testa desaparece. Pero sí, vamos a estar en contacto. Let's be in touch.
Yeah, thank you. And they just followed up and said, yes, they are North American varieties that can originally be traced back to Chile.
Oh.
Yeah.
Great.
So I think we will just turn, we had a couple more questions, but we'll put them on Slack for you to be able to look at and respond to, since we're running out of time here. And again, I just want to thank you and Daniel for all the work that you're doing to support Quinoa and for your presentations today and participating in the symposium.
Muy bien, pues le queremos agradecer a María y a Daniel por sus presentaciones. Quedaron algunas preguntas, pero las vamos a poner en nuestro canal de Slack y ahí van a poder encontrar las respuestas. Les agradecemos a María y a Daniel por su trabajo con la quinoa, por todo lo que hacen con relación a la quinoa. Y pues muchas gracias a todos.
I also want to thank Laura Lewis for a very good job facilitating this session.
Yo también le quiero agradecer a Laura Lewis por todo su trabajo en la facilitación de esta sesión.
And extend another thank you to our speakers, and also thank them for the great discussion before the session even started.
Y también le quiero agradecer a nuestros presentadores, incluso antes de que empezara la presentación, porque empezaron con una discusión muy buena sobre sus presentaciones.
So this room is now closed, but you can find the next event in room 1.
Bueno, por ahora vamos a cerrar esta sala, pero ustedes pueden encontrar el siguiente evento en la sala número 1.
Starting at 3 o'clock, so about 20 minutes.
Empezaremos a las 3 en punto, o sea, más o menos en 20 minutos.
We will have a networking discussion on indigenous seed sovereignty with Dora Tambo.
Nosotros vamos a tener una discusión de network con Dora Tambo sobre semillas indígenas.
Thank you very much, and we hope to see you in room 1 shortly.
Muchísimas gracias a todos y esperamos verlos muy pronto en la sala número 1.
Thank you, Evan.
Thanks, everyone. Thanks, Laura.
Thank you.
Gracias a todos.
Hey, so I have a question here by Evan Crane that I had already translated, and for some reason it's not appearing in the answered Q&A. But it came across like 3 times. Hello?
That should be okay, Shirley. I was just trying to repost it because Laura couldn't see it for some reason.
Yeah, I think what it's doing is the Q&A is just pushing all the questions in there. I don't know that they've been copied into Slack. Maybe that's why.
Okay, I can check Slack and see.
Yeah, because I kind of scrolled through the questions, and the questions would kind of get lost in there, but they're there. I know they're there.
Okay.
I'm going to go through all of the questions right now from Maria's talk and then copy them over to Slack.
Let me know if there's one that I might have missed, but I think they're all translated at this point.
Awesome.
Hey, Evan, thanks for being Superman there and asking